because we know that our schools are located at different parts of India, even if at some remote areas, and we have got few CBSE schools in some foreign countries. That is why the CBSE has given a flexible schedule of around 4 to 8 weeks to conduct this examination in different parts of the country. Now, this examination will be a MCQ pattern of examination, which will be case-based and assertion or reasoning based. So this MCQ of these two types, this examination will be of 90 minutes duration, covering 50% of the syllabi. The questions that will be framed by the okay. So the second is the examination date and the evaluation date will be same, and in the same night, the marks will be uploaded by the principal of the school on the CBSE portal. Now, these marks obtained in the term 1 examination will contribute to the final overall score of the students. Now, let me come to term 2 examinations. The term 2 examination is scheduled to be held in March and April 2022, and this will be covering rest 50% of the syllabi. The duration of the examination will be 2 hours or 120 minutes. The questions will be in three types. Either it will be MCQ type with case-based or situation-based. Then it may be of open-ended questions, and it may be short answer or long answer type questions. Now, suppose during the time of March and April 2022, the situation is not conducive for holding the normal descriptive type of examination. Then at that time, the CBSE has decided if it is not possible to conduct the examination, there is an alternative that CBSE will also conduct the second term examination with MCQ based question having 90 minutes duration. Besides that, I have sent the different copies to the different WhatsApp group in different subjects where suppose the first term is not held due to COVID-19. Then what is the decision in the second best term 2? Now suppose first term is held, the second term is not held. Third is both the term 1 and term 2 are not held by the due to this worst situation of the COVID-19. So in these 3 cases, what is the modus operandi, how the CBSE will going to assess the students, that I've sent to all the WhatsApp group. You can refer to that. Now, I don't want to kill any time. Now, I request on my left, you find your research person for botany is Dr. Hrusikesh Naik. He's the head of the Department of Botany in Rajdhani College, Bhubaneswar. He will be delivering his talk. Now, I will request Dr. Naik that perhaps the secretary has decided that she may come sometime between 10 to 11. Now, at that time, I may request him that to spare a few minutes because the director will talk directly to the students. Thank you very much. Now, I hand over the mic to Dr. Naik to start his deliveries. Dr. Naik, please.
Good morning, everyone. So we are going to discuss कौन करेंगे Okay, dear participants, what we are going to do, we are going to have discussions on multiple choice questions on molecular basis of inheritance. Prior to this, there is classical genetics, that is on transmission genetics, Mendelian genetics. So then came your molecular basis of inheritance or molecular aspect of genetics. So this chapter we will be having the source of DNA genetic material, structure of DNA, DNA replication, and finally how information flows from DNA to protein. So we will be discussing these questions so that our concepts would be clarified and we can in a better way explain students how to get these questions. Okay, the first one is regarding the source of genetic material. Let me tell you, before 1928, there was a tussle between two classes of people, those who favored protein as genetic material and others favored DNA as genetic material. Their proposal was like this, because protein diversity is very high in comparison to the DNA having only 4 types of nucleotides. So anyway, but this was the road that led to discovery of DNA as genetic material. So we'll be discussing. So first one is bacterial transformation was discovered by Frederick Griffith. It is in 1928. Okay. Second one, once Frederick Griffith found that there is a transforming principle which transforms the 2R to 3S. Then there was a question mark. What was the biochemical basis of that transforming principle? Or what is that chemical which transforms 2R to 3S? So a step further in that direction was carried out by MacLeod, Avery, and et al. 3 scientists, they carried out an in vitro experiment on pneumococcus. Okay, they didn't take the whole cells, rather they fractionated the components of this pneumococcus and ultimately it is so it is a very et al. They carried out their in vitro experiment and finally with the help of enzymes, they found that DNA is the genetic material. So Avery et al. Proved transforming principle in pneumococcus is DNA. But why it is pneumococcus? Sometimes it is written diplococcus, streptococcus, and pneumococcus. But pneumococcus, because it is caused pneumonia and a coccus form of bacterium. Initially, it was found to be in 2 forms or 2 spherical forms. So that was diplococcus. But when it was found that is in chain, strepto means chain. So that was ultimately named as streptococcus, and but fondly it is also called pneumococcus because it causes pneumonia. So in pneumococcus, it was proved DNA as the genetic material. Okay, but gradually, actually, I have arranged the questions this way. As the sequence of work by the scientist went on searching the DNA genetic material, so in that way, the same way I have arranged. So in 1944, Abhay et al., they communicated to the scientific world that DNA genetic material, but people, those who argued that protein as the genetic material, they could not be satisfied with the experiment of Avery et al. So that's why in another experiment, Horsey and Chase, in 1952, they proved that DNA genetic material in phases, bacteriophages, T2 bacteriophage. And we know what was the basis of this. So DNA, the genetic material in phages, was proved by, it is our Horsey and Chase. So they took T2 bacteriophage. How we can orient students from one question to many? How can we do? So what was the experimental organism? It is T2. What were the tracer elements? They were S35 and P32, radioactive forms of sulfur and protein. But what was the logic of this experiment? Actually, this was differential labeling of two batches of phages. One batch was labeled with P32, other batch was labeled with S35. And ultimately, when phages infected the host cells, the E. coli, it was the P32 that was found inside the host cells. It is not the S35. As we have read the lytic life cycle, there we have seen the ghost comprising of the protein coat remains outside. So you can have 10 questions from this. Then experiment on pneumococcus proved that bacteria do not reproduce asexually. Bacteria undergo binary fission. DNA as genetic material. So third one is the answer to this. Then RNA contains the sugar. Once we are in a position to prove to the scientific world that DNA are genetic material, but here all these three experiments proved DNA genetic material. But let me tell you how we can fine-tune students a step further. RNA genetic material, students have come across tobacco mosaic virus, which is a RNA virus. But in tobacco mosaic virus, RNA was proved to be the genetic material. So from bacteria, in bacteria it was proved DNA. In some viruses, it was proved RNA as the genetic material. So ultimately, nucleic acids are found to be the genetic material of the living world. So as it is not there in the course regarding RNA, but we have to prompt them like this. Next, regarding RNA. RNA contains the sugar. We know this is ribose. It is a pentose. So ribose sugar is this. But here we can shift the question to DNA also. DNA contains the sugar deoxyribose. But at what position? It lacks oxygen. It is the second position. But to all my friends present here, let me tell you, whenever we go for a discussion that is 3 5 like this, 5 to 3 end like this, what that dash or priming refers to? That priming of carbons of sugar, because we write 1 dash, 2 dash, 4 dash, 5 dash. It is the carbons of the sugars. Why it is so? Because the biochemists, they have given this clue that the carbons of sugars must be primed, because in a nucleotide, sugar is there, base is there, phosphate is there. So whenever we speak 1 dash, it refers to the first carbon of sugar, not that of the base. So it is a distinction. It is international nomenclature of the sugars in the context of nucleic acid nomenclature. So next we come to pyrimidine. But let me tell you here in question number 5, RNA as the genetic material in certain viruses, we know TMV, HIV, and many tumor viruses have RNA as genetic material. But all cells, that means all cellular life forms, let it be prokaryotic or eukaryotic, all of them have DNA as genetic material. Why? DNA was chosen by cells as genetic material rather than RNA, because DNA is more stable in comparison to RNA, right? And one factor behind it is this deoxyribose sugar. Let me tell you, wherever oxygen is there, oxygen causes oxidation of molecules. So ribose having oxygen, one oxygen extra in the second position, is prone to attack. But deoxyribose, which lacks oxygen in that, so that is behind the stability of the DNA, one factor. There are many factors why DNA is stable. Okay. In our cell, every cell, we have a lifetime supply of DNA. As long as DNA, we have the cell, we have the DNA. But many tons of RNA comes, or RNAs are formed and degraded. So RNA has short life in comparison to DNA. So that should be guided to the students in that direction, because if you give clue to the students, they will drag the things forward. So pyrimidine base present in RNA in place of thymine. Purines and pyrimidines, we know purines are double ring structures, pyrimidines single ring structures, and the pyrimidines unique to RNA is uracil. Okay, and that is the substitute of thymine, so to say. Then you come to this question: which of the following is most accurate sequence of intermediates during protein synthesis by an RNA tumor virus? This question, I think this is from Niger or Olympiad question that I have collected. What is the sequence of flow of information? We know the sequence of flow of information in cells, that is from DNA to protein via RNA, an intermediary. But in case of RNA containing virus, what is the sequence? So RNA is their genetic material. Once they enter their cell, if once HIV enters our cell, then that HIV carries the retrovirus, reverse transcriptase enzyme. With help of that enzyme carried, the RNA is copied to DNA, reverse transcription. Then a DNA gets integrated to one of our chromosome in an abrupt manner in T4 lymphocyte. Then from DNA, mRNA, then protein. This is the answer. The first one. Okay, because these things come in many examinations. So students should be taught conceptually. Yes. Next one. This is relating the structure of DNA, relating Chargaff's rule. Given that a nucleic acid has 30% adenine, 20% guanine, 30% thymine, and 20% this thing. So from this, what is predicted? What is the most likely structure of DNA? Adenine pairs with thymine, we know. So as per Chargaff's rule, the most likely structure is double-stranded DNA. I hope you can be well abreast with this. The correct next is the correct order in terms of molecular weight. Yes, we have all kinds of macromolecules in the cell, but how the students will take the sense of their size, molecular weight. So let's come to this. Answer would be this. The answer is B, because immediately whenever I am explaining before you, I am also giving a thinking to this. tRNA are soluble RNA. They have a size of nearly 4S, and they range in size from 73 to 95 nucleotides long. So tRNA is best, and then mRNA, because mRNA is a copy of the gene after processing. Then rRNA, they are having huge size. We have read different size of rRNA like 23S in bacteria, 16S, different sizes are there. So rRNA is next and DNA is the longest one. So this would be the answer. Right? Next. But from this, how the students can get these B, C, Z, and all these. When I was teaching in graduation, this time I asked students, final year students, what do you mean by B? You people have written the differences between B and Z and A DNA. But what is B? B is biologically active DNA. Because DNA is localized within the nucleus or nucleoli in a cell. In well-hydrated cell, the DNA that remains, that is your B DNA. B stands for biologically active DNA, and Z stands for zigzag nature of the sugar phosphate backbone. Zigzag nature of the sugar phosphate backbone. But in B DNA, it follows a smooth pathway. Okay. But you have to invest more time on this. Melting of DNA is the process of separation of the complementary strands by heating. So following DNA molecules should melt at the lowest temperature. So how you can give? Answer would be those having the number of base pairs are fixed in all. Number of base pairs are fixed in all. But thing is, how many GC pairs and how many AT pairs are there? So those having highest number of AT pairs would melt fast, because A and T, they are bonded by 2 hydrogen bonds. So GC rich DNA melts at a higher temperature and AT rich DNA melts at lowest temperature. That is the basis. That is the clue. So the students have to see how many total base pairs and how many. I think in this there is highest number of AT pairs than others. Others have more of GC pairs. So this would be the answer. Answer C. And answer to all these is given in the last. You can follow that. Which of the following statements about the base composition of a double-stranded DNA molecule? Okay. So A is equal to T within each single strand. C is equal to G within each single strand. A plus T is equal to C plus G in double stranded DNA molecule, A by C, A by T. So this would be the answer. So actually, we will mark in many there are 4 options, in many there are 5 options. Actually, the 5 options in each question, wherever they are there, they are very tricky questions and collected from either national standard examination in biology. So that is NACV, it is coming, Olympiad, Niger, somewhere from these questions are collected. So our basis is A is equal to T in each single, there is no parity. The same also here. A plus C is equal to C plus G. It is not also. Always we have to adhere to the Chargaff's rule. The molar concentrations of adenine is to that of thymine, and molar concentrations of cytosine is same as to that of guanine. But AT, number of AT pairs and GC pairs in DNA varies. So this one also is not the answer. So A by T okay, we'll see this. What will be the answer? But first one, second one, they won't be the answers. Third one, 80 pairs is equal to GC pairs in double stranded DNA. It's not, but AC is not. So there will be some typological mistake. I will see this and clarify it towards the end. No problem, but we will not invest more time in this. So which one of these is true for DNA? You see, AT, GG, and this is very simple. This one, third one, rest is not. This speaks the pairing principle. Adenine, thymine, this is GG, but this one. Answer is C. Which one of the nucleotide compositions will be possible if DNA is double stranded? Okay, I have already made bold it. The answer is this. Okay. The 2 polynucleotide chains in DNA are antiparallel. But with this, I am not going to explain what antiparallel nature is, because we know students in DNA strands, if we write the 2 strands are antiparallel, one runs in 5' to 3' direction, the other one is 3' to 5' direction. Acha mu eta ku minimize kari, I can go to PowerPoint. Oh, okay, I will be coming. Because in the 2 strands of DNA, what determines the antiparallel nature? It is the arrangement of sugars. If you see the sugar is 5 carbon. Sugar is 5 carbon. So it is a ring structure. In ring, there is one oxygen at the top. So it is the puckering of sugars in one chain. The sugars with oxygen in one direction, in the other strand, that is in downward direction. If possible, I will show. I have a PPT having some pictures, only pictures. From that, we can be clarified this, what is antiparallel nature of DNA. And this antiparallel nature is determined by the sugars. Then nucleotide arrangement in DNA can be seen. When I saw this question yesterday night, then I saw nucleotide arrangement. It is sometimes may come nucleotide or sequence of base. It is not as speaking of the sequence of bases, but how the nucleotides are arranged in DNA. Is it ultracentrifuge that determines the size determines the size basing on their weight or density. Then light microscopy, it takes the pictures. Electron microscopy also, and DNA appears as a thread. But what it determines the arrangement? It is X-ray crystallography developed by Rosalind Franklin. From this, they inferred it is helical and there is repeating structure, or nucleotides are repeated, and it is of uniform diameter. Three things, that is of uniform diameter and helical and repeating. So these three things were inferred from the X-ray crystallograph developed by Rosalind Franklin and Maurice Wilkins. Next, if the sequence of bases on one strand of DNA is given as this, the sequence, this is simply complementary. But thing is, in this end, this is G, so definitely it would be C. Wherever C, this one, immediately this comes. If this is G, this would be first would be C. Then this is C, then it would be G. No, there is somewhere reverse. There somewhere reverse. You can see this. So it is just simply the complementarity of bases that can be followed. And there is some tricky thing somewhere you can read. The year 2003 was the 50th anniversary of structure of DNA by Watson and Crick, because it is 1953 that gave birth to the molecular genetics, the structure of DNA. Okay, and the two stalwarts, those who didn't go for any experiment, rather they extrapolated data from Maurice Wilkins and Rosalind Franklin, this X-ray crystallography, because these people visited their lab and they found the Chargaff's base pairing principle or molar concentration ratio is very useful in determining the structure of DNA. But the helical nature of DNA, how they imbibed this idea? Actually, it is Linus Pauling, the great protein chemist, 2 times Nobel laureate, who proposed DNA might be a triple helix, not double helix, but helical nature of DNA was given, the idea was given by him. So helical nature from Pauling's idea and other data from Maurice Wilkins and base pairing principle from Chargaff helped entirely to helped their wise thinking in developing DNA agenda. So you see, people experimented, but who own these three? And Chargaff has lot of resentment. In his essays, he has written, I am much neglected, because he didn't get any recognition in this regard, because his data was used by others for extrapolating and formulating DNA double helix. Anyway, so this is quite interesting because this is a Nobel Prize, and not only Nobel Prize, but that structure revolutionized molecular biology. Okay, this one, telomeres. When we had our MSc course, we didn't get this term telomeres. Okay, because during 90s, the concept of telomere came. So each eukaryotic chromosome is linear, having 2 telomeres, 2 termini. So what that does, it controls the function of eukaryotic chromosome. It controls the function of eukaryotic chromosome. How? Prevent chromosome loss, act as replications, help chromosome pairing, or RNA transcription initiator. Actually, Elizabeth Blackburn, who discovered this telomeric sequence and the role over a span of 20 years, there is a lot of discussion on telomeres. So telomere is just like a cap. So two caps at two termini, two ends of eukaryotic chromosome. So they prevent chromosome loss. They prevent chromosome loss. We must have heard Thomas and Morgan, and another is our McClintock. When they found there is chromosome breakage. There is chromosome breakage. That means chromosome ends are lost. The chromosomes with lost ends, they fuse. Two different chromosomes, irrespective of anything. If they have lost their ends, they fuse. So their discovery helped Elizabeth Blackburn to come to a conclusion that these telomeres are essential for maintaining the integrity of the chromosome. Okay, and telomere is, we will see, that is a repeat sequence. Not only it prevents chromosome loss, it has many functions. Many functions. I will be coming to that. Okay, Escherichia coli. One thing, E. coli, Escherichia coli, this was discovered in 1885 by Theodore Escherichia from the colon of human beings. Actually, he found this bacterium from the human feces, and the human feces are stored in the colon. That's why, as it was discovered by Theodore Escherichia, the generic epithet Escherichia and colon inhabiting, that's why coli. So Escherichia coli, this is regarding the DNA replication. But one thing, how you can impress students, how can drag students towards your own. E. coli is the most thoroughly studied organism in this earth. No organism is so thoroughly studied in terms of genetics and metabolism as E. coli is. Okay, so because E. coli is easily isolated and easily grown and not infectious, this strain especially. So it is fully labeled with N15. We have, once we understood the DNA double helix, then came that how the genetic material, the DNA replicates. One DNA gives rise to two DNA, that is replication. So to prove the semi-conservative mode of DNA replication, because three modes were proposed, but what ultimately proved, that is semi-conservative mode of DNA replication. I have some diagrams and I will show you after just having 30-40 questions. So it was N15 nitrogen, because nitrogenous bases are present in the DNA. So it is N15 heavy nitrogen and N14. One thing you remember, these are not radioactive. N14 having 14 neutrons, and this is, neutron and proton. So it is having a mass 14 and it is having a mass 15 because it is having one extra neutron. But this extra neutron different, that led to different in densities of the DNA having many bases and many nitrogens. So when we speak nitrogenous bases or simply bases, do you know why they are called as nitrogenous? Many might be knowing, but nitrogen present in the purines and pyrimidines, they have their lone pair of electrons. These lone pair of electrons present in the ring of purines and pyrimidines attract protons, have basic behavior. So that's why the purines and pyrimidines are called nitrogenous bases or simply bases. They have basic behavior. Okay, the two strands, this one. Okay, so what we got from this? What is our basis? When our DNA having all nitrogen as N15, another having all nitrogen as N14, they could be centrifuged in a density gradient centrifugation as two different bands. And actually this one because of this. Anyway, so different density and do not resemble parent. Actually, after one generation, transferring the heavy nitrogen, heavy DNA to light DNA, light medium, then after one generation, they have a different density and do not resemble parent DNA, different density but resemble parent DNA. How you can differentiate? They have same density or different density? They have different density because heavy DNA, light DNA. But after one generation, that becomes your hybrid DNA. Each DNA molecule having two strands. One is parental strand with N15, the other strand with N14. So that occupies a band intermediate between the heavy and light. So that has a different density and do not resemble parent DNA. Different density, but resemble parent DNA. This one, second one is the answer. Okay. Next, if double-stranded DNA with the N15 undergoes replication twice in normal medium. Twice, because after one round of replication from one DNA, two DNAs are formed. Then after two rounds, 2 to the power 4, 2 to the power 2, 4. So 4 DNAs are formed. So out of 4 DNA molecules, half of the bacteria contain N15 in DNA. All 4. So which one would be the answer? So half of the bacteria contain N15 in DNA. Because after 2 rounds of DNA replication, there will be 4 DNA and 2 will be having this hybrid feature and 2 having completely new feature, N14. So that's why 50% having hybrid feature and 2 are having new, that is N14. So this would be the answer. Then this is regarding replication, because once we understood that DNA replication is semi-conservative, and that was proved by Messerson and Stahl by using density gradient centrifugation in 1957. So then came the mechanism, and we know the Kornberg enzyme, that is DNA polymerase. So gradually how the sequence of scientific findings has come. So the enzyme that catalyzes the elongation of a DNA chains in 5' to 3' and is template dependent called polymerase. Okay, that is polymerase, DNA polymerase. Messerson and Stahl in their experiment of DNA replication use the technique of, so organism is E. coli. The technique is density gradient centrifugation, in which they created a gradient from light to heavy cesium chloride, because cesium is most metallic and most heavy salt. So cesium chloride solution was in a density gradient manner from top to the bottom, light to heavy. That gradient was created. So accordingly, density gradient centrifugation was used by these two scientists for proving proving semi-conservative DNA replication. Even though 3 modes were proposed, but what ultimately came true, that is semi-conservative mode. Next. Okay. Cells were cultured in a medium containing heavy isotope. This is a lengthy one. Cells were cultured in a medium because once we put our brain heavily, then we can know. Cells were cultured in a medium containing heavy isotope of phosphorus, P32, till the entire DNA complement had only this isotope. Subsequently, some cells were transferred to a medium with a normal isotope, P30. After a certain period, some cells were harvested and analyzed for heavy phosphorus. It was found that only 6 to 6.25% of the phosphorus in DNA was heavy. How many times the cells must have? You have to put your brain here. So it is 4 times, because a DNA on 4 round of replication yields 16 DNA molecules. Out of 16 DNA molecules, 2 are heavy. Actually, 8 times এটা को calculate କରିବାକୁ ପଡିବ, but this is a quite interesting question. A teacher has to put his brain first on this. A lengthy, this is not of a CBSE question, this is a question from this Niger. Heavy phosphorus containing DNA was transferred to light medium, not heavy, that is radioactive phosphorus containing DNA was transferred to a medium with normal phosphorus. So after how many rounds of DNA replication, the DNA having only P32 was 6.25%. So that is 4 times. 2 to the power 4, 16. So replication of DNA in eukaryotes. But prior to this, let me tell you, replication of DNA in eukaryotes begins at. This is a question which seems to be absurd. But prior to this, let me tell you, prokaryotes have a single circular DNA. At how many places replication starts? It is 1. That is ORIA, that is origin of replication. So prokaryotes have a single origin, and prokaryotic DNA replication is faster. Somewhere will be, yes, here. We'll see that in prokaryotes, DNA replication is faster, but in eukaryotes, DNA replication is much slow. So to cope with much slow rate of replication and long DNA molecules, eukaryotes have 1 or more, actually many replication units. The answer would be 1 or more specific sites on a chromosome and proceeds bidirectionally. The first half is okay, 1 or more specific sites and proceeds unidirectionally. DNA replication, irrespective of the cells, prokaryotic or eukaryotic or viral, whatever, it is bidirectional replication. So this one is the answer, 1 or more. Let me tell you, the site from which DNA replication starts in eukaryotes, that site is called autonomously replicating sequence. But in prokaryotes, the same region, the same sequence, that is called ORIR. That means origin of replication. Then rate of addition of nucleotides per second, that means the speed of replication they are telling. So DNA polymerase 3 is the most important polymerizing enzyme, but its rate of addition or rate of polymerization, that means it adds 1000 nucleotides per second. So it is very fast. Next, but when we will be comparing this prokaryotic DNA replication with that of eukaryotic, it comes to around 50 nucleotides per second in eukaryotes. During DNA replication, the bases of two strands separate by because DNA helix, the two strands are held with each other by means of hydrogen bases existing between the hydrogen bonds existing between the bases of the two strands. So the two strands for separation, the enzyme that acts to break the hydrogen bonds, it is helicase, and helicase requires ATP. So the separation of strands at the origin and ahead, I mean, wherever there is separation of the two strands, helicase works, and this is ATP dependent. Yes, this is ATP dependent. Otherwise, if it is ATP independent, no energy is required for breaking. Anytime, any moment, the two strands can separate. No, but the DNA is very stable. I say most stable molecule among all. So DNA replication is aided by. We know DNA polymerase, but is it the only enzyme? Actually, DNA replication is a very complex process, but it is well understood now at this time. So who are they? At least from our previous knowledge, we know the DNA polymerase and DNA ligase. They are essential for these two. So answer would be both A and B, this one. Semi-conservative mode of DNA replication using N15 was demonstrated by Messerson and Stahl, already discussed. Semi-conservative replication of DNA was proved by using, somewhere repetition is there, and that is N15. N15. But this one, P12, there is no P12. It is P30 or 32. Similarly, S32, S35. N14, N15. C12 is there. But this is our nitrogen 15, N15. Which is happening. I will take a little tea. Dear participants, actually, it is we. Let me tell you, whenever we go to any refresher course, because I am a teacher, I also go for a refresher course for my career also. Everywhere, people think that we know this, we can do this, but whenever these questions are put before you or before me in a hard copy, we fail to answer many things. It takes a lot of time. So that's why it is my request, be analytical to these questions. What I am speaking, try to listen and give the feedback, or you can have an interaction well. To form a continuous DNA molecule, the enzyme that joins, or the enzyme that joins Okazaki fragments is your DNA ligase. Because if I ask, this enzyme has a EC number, please cross-link this. This is an enzyme, ligase. Enzymes have six classes. So to what class this ligase belongs? So its enzyme commission number should start with what? 1, 2, 3, 4, 5, 6. If I give this question, immediately you have to answer. This class 6 enzyme. Then DNA polymerases are generally used in DNA replication to cut the helix at certain places, adding carbonyl compounds for proofreading, joining pieces of a DNA strand. Yes, DNA polymerases are generally used in DNA replication. Many options are there, but from this, what you can find? To cut the helix at certain places, no. Adding carbonyl group, no, because carbonyl group is not there in. For joining pieces of DNA, no. This joining DNA pieces is the job of DNA ligase, but we are speaking of DNA polymerase. So for proofreading, whenever there is polymerization, addition of new nucleotides, when we are writing, there is possibility of mistake. How the mistakes can be corrected if we read this and proofread this. So writing, then reading will help us correcting our mistakes. Similarly, DNA polymerase 3 is bifunctional. It is a polymerase as well as exonuclease. So proofreading activity would be the answer to this question. Semi-conservative replication was first demonstrated in already discussed E. coli. Then the main enzyme responsible for new strand synthesis during DNA replication in prokaryotes, that is DNA polymerase 3. 3 polymerases are there: DNA polymerase 1, 2, and 3. Okay, let me divert little. Which one of these is called Kornberg enzyme? Arthur Kornberg discovered which enzyme? That is polymerase 1. Polymerase 1. The story is like this: DNA polymerase in terms of size is huge. So during the process of isolation, this was captured first or caught first. But subsequently, towards 1972, DNA polymerase 3 was discovered. Once these other enzymes relating DNA polymerization were discovered, they were given the Roman numeral 1, 2, 3. Okay, but DNA polymerase 3, which is present in very low number in the cells, but that is most pivotal, most important enzyme. This is the real polymerizing enzyme. But we will be coming in other questions to the role of DNA polymerase 1. Both have the role, but this is the major polymerizing enzyme. This polymerase has polymerizing activity as well as proofreading activity. So proofreading is done because of 3' to 5' exonuclease activity. Okay, it's fine. This question I have highlighted, making this red. During DNA replication, the synthesis of lagging strand involves the activity of following enzymes, because there are 2 strands. One is continuously synthesized strand, is called leading strand. The other strand, or the other nascent strand, that is synthesized in patchy manner, that is your lagging strand. Because leading strand is so called because the growing end of the continuous strand is much ahead of that of the lagging strand or discontinuously synthesized strand. Sometimes it comes to our mind why it is called leading strand. Continuous strand we can easily understand, because one is synthesized continuously, the other one is discontinuously synthesized. But continuous strand is called leading strand and discontinuous strand is called lagging strand. If you see the growing end of continuous strand and the discontinuous strand, the continuous strand have the growing end much ahead of the discontinuous strand. That's why it is called leading strand. It leads out of the 2 strands, it leads. That's why leading and lagging. Next, in the lagging strand, here the question is centralized to this DNA replication. Synthesis of a lagging strand involves the activity of the following enzymes. All these 4 are involved. What is the sequence? You see here, which one would be the answer? The first one, because wherever DNA replication is to start, it must be primed, because a short segment of RNA would provide the growing point. So this one would be the answer first. If option 4 thibo, that would be first. You see, RNA primase followed by DNA polymerase 3, that is 2, this one answer. because 4 is RNA primase. This will form a primer. DNA polymerase 3 will extend from that, or it will go on polymerizing. And third, this is 1. You see, 1 role konhala DNA polymerase. Somewhere I can explain with the diagram also. DNA polymerase 1. DNA polymerase 1 carries out nick translation. Remember, it is nick translation. Nick translation means the DNA polymerase 1 removes the RNA primer of the Okazaki fragment ahead. It removes by exonuclease activity and fills the gap. It does 2 functions: DNA polymerase 1, which is a exonuclease, as well as polymerase. Okay, then once the gap is filled, then comes the function of a ligase. So this one be the answer. Because when this type of question comes in the examination, entrance examination, unless students understand this well, it would be difficult. So the Okazaki fragments in DNA chain polymerize in 5' to 3' direction and explain 3' to 5' replication. So wherever DNA replication takes place, the enzyme's property is to proceed from 5' to 3' direction. 5' to 3' direction. And in discontinuous strand, DNA replication proceeds an overall 3' to 5' direction. If you see the diagram, you can understand. In the beginning, I told you DNA replication is a very complex process. And E. coli has been a model in explaining all the questions relating DNA replication. Then, this is about DNA replication to some extent, because all the facts or concepts relating DNA replication cannot be explained in terms of questions. But next we come to function of a gene. Function of gene. This comes Biddle and Tatum. George Biddle and Edward Tatum are known for the discovery of one gene, one enzyme hypothesis. You see, this is in 1940s. These two biochemists, what they did, they discovered that by mutation study of mutation in this Neurospora crassa. Next question. Okay, in Neurospora crassa, which is a haploid, and easily mutation can be induced and biochemical analysis can be carried out. Okay, so by taking Neurospora crassa, they established one gene, one enzyme theory, for which they bagged the 1958 Nobel Prize in Physiology or Medicine. Okay, if you look at the questions, the sequence. Once DNA replication, then we come to gene. What a gene is, what a gene does. This is exactly what a gene does. Gene produce enzyme, right? The idea that genes control the production of enzyme. Listen, question number 37. These two scientists, they proved that genes produce enzymes. But if you go back to 1908, when the molecular genetics or molecular biology was not born, to, so to say, because DNA, genetic material was not known, structure of DNA was not known. So at that time, this British physician, who is a doctor, he studied inborn error in metabolism, Archibald Garrod. Actually, by studying these diseases like phenylketonuria, his conception was like this: why these patients have defect in their metabolism, and these defects must be at the level of genes. Even though of what genes are made of, it was not known, but his perception, his idea that genes control the production of enzymes was fantastic. So actually, he wrote the book Inborn Errors in Metabolism. So metabolic errors are inborn, and that is genetic. That was a famous book. Anyway, so Tatum, who is related to this work, he is not, but the person that is Garrett is involved in this idea. Next, the enzyme in some RNA viruses which has RNA template to synthesize DNA. Actually, RNA viruses, for example, here the tumor virus and your retroviruses, exactly the RNA containing viruses which reverse the flow of genetic information from RNA to DNA. They have a specific enzyme in them, and that is your reverse transcriptase. If we normally analyze flow of information in a cell, that is from DNA to RNA, then to protein, that is flow of information. But these viruses have the unique property and unique component, that is reverse transcriptase. So reverse transcriptase is a polymerase. What it polymerizes? It polymerizes the DNA. But what it uses? It uses RNA. RNA as a template, but synthesize DNA. So the enzyme in some viruses, the answer is the enzyme is reverse transcriptase, which is RNA dependent DNA polymerase. If we compare this with RNA polymerase is DNA dependent, but RNA polymerase. But this one is RNA dependent DNA polymerase. Okay, then gradually we are coming to genes. The protein coding part of a gene, we'll see. Gene is a stretch of definite stretch of DNA in a chromosome. It has specific length. As our house has different compartments, similarly a gene has also different regions. But here the question comes: the protein coding part of a gene, that is cistron. The functional region of a gene which is meant for producing a protein is your cistron. Okay, because it is Seymour Benzer in 1957 who coined the term like mutation. He spoke that the gene comprises of three parts: unit of mutation, which is called mutant, and now that mutant is every nucleotide is a mutant or unit that can be mutated. Recon, unit of recombination, the unit that can be recombined. So it is a nucleotide or few nucleotides that can be exchanged between homologous chromosomes. And cistron, that is the functional unit. Out of three terms coined by Seymour Benzer, mutant, recon, and cistron, the cistron came true. That is the functional part of this. Okay. Then we will be coming to exon, because whenever we teach students, eukaryotic genes are split genes. They have exons and introns. Are we in a position to tell that what exon is and what intron is? Intron is that which is not expressed in or which is not present finally in the mRNA. So exons splice back to form the messenger RNA. that we speak. But one book that I read, right, that has explained what exon and intron, how the terms came. Terms are not biological, terms are not absurd. They have their definite origin. When the split nature of eukaryotic genes was revealed, and it was seen that the regions which express, if you see, if I make this expressing region, you can see X from expressing region ru aschi ON. So expressing region is shortened to exon. Similarly, intervening region or the regions or DNA sequence which intervene the continuity of exons, that is, these are called as introns. So introns come from intervening regions, exons come from expressing region. So once you understand this well, you can feed students accurately. So students won't fall to confusion. Okay, in split gene, the coding sequences are ultimately the exons. Okay, in split gene, the coding sequence is exon, because these exons splice back, or after removal of introns, the exons are joined back to form the continuous sequence, and that leads to the cistron of the gene. Okay, the segment of DNA. Here we can just compare. Prokaryotic genes are continuous, eukaryotic genes are discontinuous or split genes. You won't find such exons and introns in prokaryotic genes. And further, in prokaryotic genes or prokaryotic chromosomes, many cistrons, protein encoding regions, are under the control of a single promoter. So that's why that is called polycistronic. Whereas in eukaryotes, a single gene or a cistron is under a single promoter, or every cistron has its own promoter. That's why they are called as monocistronic. But prokaryotic genes are polycistronic. Okay. Okay. Now, this question is relating gene. Very often we tell gene is DNA, DNA is gene. But I won't agree. Gene is DNA, definitely. Gene is DNA, but DNA is not a gene. Fact-wise, I am telling, whatever amount of DNA we have in our genome, only 3% of that, or nearly 3% of that DNA is genic. That means genes comprise only 3% or below that, or nearly 3% of DNA present in our genome is genic DNA. Rest DNA does not code for any functional product. So that is non-genic DNA. Most important, 97% of the DNA present in our genome, that is 22, in case of human beings, 22 autosomes and XY, these 24 chromosomes comprise human genome. So whatever length of DNA is present in that, only 3% of that occupied by gene, right? So what a gene is? Chromosome 1 contains nearly 2000 genes in ours. But a gene is a stretch of DNA. Gene is a stretch of DNA, which codes for a protein. Gradually, I am coming to that. So there is much non-genic DNA. Remember, much non-genic DNA in eukaryotes. Then, is it gene that codes for protein only? When we go for defining a gene is a stretch of DNA which codes for a protein. That's true, but we have left some part of that. From what source tRNA and rRNAs come from? So tRNA have their corresponding gene, rRNAs have their corresponding gene, proteins have their mRNA and have their corresponding genes. So protein encoding genes, tRNA encoding genes, rRNA encoding genes, all are genes. They are of different sizes. They give rise to 3 classes of RNA, right? So gene is a stretch of DNA which codes for either protein or tRNA or messenger RNA. So we should explain students like this. Then, which of the following elements is not involved in the information transfer from DNA to finished protein? That means when protein is synthesized, protein is okay, we'll resume. So it is what component not involved in protein synthesis, and that is your DNA polymerase. Rest 3 are involved. The process of flow of information, question number 45. The process of flow of genetic information from even while studying biology, many students get afraid of this. But tell students that is term based, word based. Here, two processes, transcription and one is translation. Simply explain students that during transcription, DNA is coupled to RNA. No kar debi na, yes. Okay. Okay. So DNA no. While DNA is giving rise to RNA during transcription, why the term so called? DNA and RNA both are nucleic acids, both are written in the sequence of bases, but DNA has ATCG, RNA has AUCG, but language is same in both the cases. Script is changed. As we say Kotiki Odia or Kotiki Bhasha, Sambapuri Bhasha, Western Odisha Bhasha कहउछन्ती, Hindi रे सेमती different scripts are there. Devanagari, Bhojpuri scripts are there, but language is one. Here DNA and RNA both language is same, scripts are changing, little change. So as in this process DNA is copied to form RNA and script is changed, that's why the term transcription is used. But translation, the process in which information in mRNA changes to the sequence or information of bases in mRNA changes to amino acid language. Language is changed. That's why it is called translation. And you people are doing translation during your school days. Okay. So that way you can explain why these two terms are so named, transcription and translation. Once students get the fundamentals, they will be attracted for the subject. Never they will get away of this. Then, all of the following are the types of post-transcriptional processing of RNA in the nucleus, except, okay, this question is relating to eukaryotic post-transcriptional event. And we know eukaryotic genes are split genes having exons and introns. The first RNA formed from DNA during transcription, that is called primary RNA transcript, that contains both exons and introns, right? So we know exons must be put back together by removal of introns. Now the question asks, all of the following are the types of post-transcriptional processing of RNA in the nucleus except which one? The removal of intron segments, polyadenylation, capping, and nucleotide methylation. Removal of introns is splicing. Polyadenylation, that means addition of nearly 300 adenylate nucleotides at the 3' end of the mRNA. So this is the first, this one is called splicing. This one is your tailing, addition of tail, poly tail at the 3' end. And this one is addition of 7-methyl guanosine at the 5' end of mRNA. So all these three are post-transcriptional events, but nucleotide methylation, that means addition of methyl group to any nucleotide, is not considered to be the post-transcriptional processing, right? But methylation happens. But here methylation is not part of this processing event. Which one of the following sequences of DNA could code for a stem loop structure? Yes. Here you can see stem and loop, tRNA. In an arm, there is a stem and loop. Stem is paired region, loop is unpaired region. And from this you can see, if this has GGCC, then here you can see CCGG. So there is possibility that this one, in the first case, it will form stem and loop, because it depends on pairing and unpairing of the RNA formed. Okay. Which of the following sequences of a DNA could code for a stem loop? Code for means this will give rise to an RNA. Would that RNA remain single-stranded or will form stem and loop? You must be remembering the arms of tRNA. DHU arm, T-Cy arm is there. There is stem is paired region, loop is unpaired region. So wherever complementarity at the both the ends exist, that will give rise to stem region, and unpaired region is loop. The basic fundamental is this, but you have to work out this. The flow of information from DNA to mRNA and then to protein is called what? Because both are there. DNA to mRNA, then to protein. That means transcription and translation together. This one is gene expression. Gene is nothing. Gene is an information center, but gene expresses itself in the form of protein. That involves transcription, the first one, translation, the second one, but in toto, that is called gene expression. The part of a gene that contains complete information for a polypeptide is, that is cistron. Replicon is there, that is a replication unit. Cistron, that codes for a polypeptide. Mutant, I told you earlier, mutant and recon. The diagram shows an important concept of genetic implications of DNA. Fill in the blanks A to C. DNA to mRNA, then protein, then protein. Who proposed this? A is transcription, B is translation, and who proposed this? It is none other than Francis Crick. Francis Harry Crompton Crick, FHC Crick, who proposed the central dogma concept in 1957. But that was subsequently proved that each step occurs therein. Right? 4 scientists, Erwin Chargaff's rule, that is base composition of DNA, James Watson, who is behind the DNA double helix structure, Rosalind Franklin, X-ray crystallograph, but Francis Crick alone proposed this central dogma. 51. DNA elements which can switch their position are called yes. While I was coming, I was thinking how to put a question relating transposon. Yes, there is. Transposons are transposable genetic elements. So transposons are sequences, short sequences, which can transpose themselves anywhere in the chromosomes. This has a long history. Transposons were discovered by Barbara McClintock, the lady scientist who worked with compound microscope, simple compound microscope, very primitive type of microscope in 1950s, because nowadays very higher versions of these microscopes have come, but with a microscope alone, she could discover these transposable genetic elements which were initially named as jumping genes in maize. If you see or simply go to the net and you search for jumping genes, and only maize photos come, maize with different grains of different colors, they come. And that the maize we are eating, it is usually of all grains, all offsprings are of the same color, but the maize in which she worked, that had variations in color, and that was because of the transposable genetic element, initially known as jumping gene, and she was given a Nobel Prize in this field. So transposons are genetic elements which transpose themselves arbitrarily elsewhere in the genome without any definite consequence, whatever consequence may be after transposition. So that's why transposons can alter or change the linearity of the genes in a chromosome. Amar eibhale chinta thai, all genes maintain a fixed position in the chromosome, but 1, 2, 3, 4, 5, 6, 7 like this, it is continuous. If one gene from other chromosome is transposed between 2 and 3, definitely that sequence is changed. So transposons change their sequence. So it is very important concept, and the initial transposon she discovered that is ACDS element. So many questions are coming relating this transposition, and in tDNA, transfer DNA of agrobacterium that we are teaching in biotechnology, the tDNA is bordered by two ends, that they are the two transposons. Because of the two transposons bordering the transfer DNA in TI plasmid of agrobacterium, it is able to transfer to eukaryotic genome. Remember, whenever I do tell you that transposon is a story, Nobel Prize is associated with this, where these things are involved. Simply reading these questions will hardly give any concept. Try to correlate this. During transcription, the strand not transcribed by RNA polymerase, right? That is, out of the two strands, the strand that is not transcribed, which one? Definitely question immediately comes to mind, that is coding strand. If you go to the books, that is your non-coding strand. Answer is non-coding strand. The reason being, out of two strands, the strand that is copied to synthesize RNA, that is called template strand, no doubt. That is called template strand. But here question comes, the strand not transcribed, that means non-template strand nanta kon hobo, because many times students, teacher go on arguing on this. That is transcribed is the template strand, and that is called non-coding strand, right? Sorry, the strand that is transcribed is template strand, but the question here is the strand that is not transcribed, which is not used as template. What can be the name given to that? That is your coding strand, because the two DNA strands are complementary. As the mRNA is in sequence is as good as the non-template strand, so the non-template strand is called coding strand. Okay. Central dogma. Central dogma was proposed by Francis Crick, that is already discussed. The process by which DNA of a nucleus passes information to RNA, that is transcription, no need. Genetic information in DNA molecule is stored in the form of, yes, sequence of amino acids, sequence of nucleotides, sequence of nucleosides, and sequence of sugars and phosphate. Actually, we know it is the sequence of nucleotides, and more specifically, it is the sequence of bases. So third one is the answer. It is not nucleoside, right? And sugar and phosphate, if you explain students, sugar and phosphate, they form the backbone of the DNA. But what is the variability of DNA? Wherever you may bring DNA from virus, plant, or animal, in a DNA molecule, in a DNA strand, single strand, sugar phosphate is constant. So that is the constant feature. What is the variable? It is the sequence of bases. So that's why it is the answer is sequence of nucleotides, right? Which one of the following does not follow the central dogma of molecular biology? Or which deviates, the concept proposed by Francis Crick. All these are cells. You see Chlamydomonas, an unicellular alga, P. mucor, which is a fungus, but HIV. HIV has RNA genetic material and it reverses the path. Okay, so after discovery of reverse transcription, the central dogma was modified. So this would be the answer to this. Transcription began when one of the following enzymes. Now, what a gene is? Gene is a segment of DNA. Further, a gene has two different regions at least. One is promoter, the other one is coding sequence. And promoter is to the upstream of the coding sequence, 5' end of the coding sequence, right? So transcription begin when the enzyme RNA polymerase binds to the promoters. And in the book, it is written RNA polymerase, with the help of the sigma factor, is able to locate, bind the gene that is to be transcribed. So RNA polymerase is then further described as a sigma factor and core enzyme. Core enzyme is responsible, because in the book, NCERT book, it is like this. Core enzyme is responsible for catalysis, polynucleotide synthesis, RNA synthesis. But for transcription to start with is associated with sigma factor. Once transcription begins, the sigma factor dissociates and the core enzyme carries forward the process of polymerization, right? If I say DNA polymerase and RNA polymerase, DNA polymerase is not sequence specific. Any DNA sequence can be replicated by DNA polymerase, but RNA polymerase is not for any DNA sequence. It is specifically for the gene itself. They act on the gene, they act on the promoter, then carry out transcription from the coding sequence. So next is, in eukaryotic cells, the RNA transcribed from DNA is called, okay, in eukaryotic cells, the RNA transcribed from DNA is called rRNA. No. Why? Because all kinds of RNA are formed from DNA by way of transcription. So we cannot write RNA, rRNA. This cDNA, small cDNA, it is complementary DNA. Complementary DNA, that is from RNA to DNA. That is not. Then cistern is a unit, but this is heterogeneous mRNA. Heterogeneous mRNA. We can, because this term should be understood well. Why the term heterogeneous RNA or heterogeneous mRNA is coined? If you look at the gene is having promoter and the coding sequence. The coding sequence is having exons and introns. So the primary transcript has exons and introns. And we came across the term like addition of cap, addition of tail, and splicing. So in a nucleus, some RNA are just formed without processing. Some are under the process of processing. Some, that means they have a different length. mRNA is very short in comparison to the primary transcript. So the primary transcript is of much longer in comparison to mRNA. So different sized RNAs exist during the process of transcription, post-transcription. So that's why population of different sized mRNA is given the name heterogeneous messenger RNA. Then molecular basis of organ differentiation depends on the modulation. This was a question of AIPMT. Molecular basis of organ differentiation depends on the modulation of transcription. Actually, when we speak of transcription, immediately comes to our mind, in case of bacteria or in case of eukaryotes, there is an enzyme called RNA polymerase which carries out transcription. But is it aided by, is it associated by or guided by some other factors? Exactly. It is transcription initiation factor. So the answer is, along with RNA polymerase, other transcription factors which guide RNA polymerase to bind what enzyme? Sorry, what gene? So the answer is transcription factor. What we know about DNA replication, transcription, translation, it is a simplistic approach, but in reality, it is much more complex at regulation level. Okay. Prokaryotes have only one type of RNA polymerase, but in eukaryotes, there are three types of RNA polymerases: polymerase 1, polymerase 2, polymerase 3, right? How I remember that, or how we can guide students to remember which type of RNA polymerase produce what kind of mRNA? Sorry, what kind of RNA? We know rRNA, mRNA, tRNA, but which one goes for, or which one is produced by which kind of RNA polymerase? Polymerase 1, 2, 3. I have remembered RMT. Polymerase 1, RNA polymerase 1, it produces rRNA. RNA polymerase 2 produces mRNA. Polymerase 3 produces tRNA. There is a little deviation, but this is the normal. 1, 2, 3, RMT. Right? So, major difference between prokaryotic and eukaryotic polymerases are: prokaryotic have one type producing all kinds of RNA, but eukaryotes have three kinds of RNA polymerase. They have their distinct roles. They have their distinct roles, right? Polymerase 1, then if RNA polymerase 1 produces rRNA, then where it is localized? Question tapar asba taku relate kariki. RNA polymerase 1 is present no doubt inside the nucleus, but specifically at what place? It is nucleolus. It is nucleolus. You see, teminism. Teminism is the same as reverse transcription, because it is Howard Temin and David Baltimore in 1969 who discovered that reverse transcription is the case in case of tumor viruses, retroviruses, right? So, as Howard Temin is the first author, that's why Teminism is the common name that describes reverse transcription. Then, an alteration. Okay, let me stop here for 1 or 2 minutes. How the three kinds of RNA polymerases were discovered? Because in prokaryotes, only one type, but in eukaryotes, there are three types. All and sudden, how they could be known that there are three types of RNA polymerases? Actually, there is a very interesting story. We know Amanita phalloides, which is a mushroom that is called death cap mushroom, poisonous mushroom. And the active principle of that mushroom is alpha amanitin. Alpha amanitin. Alpha amanitin acts differently on these RNA polymerases. What I remember, RNA polymerase 1 is fully sensitive. RNA polymerase is partially sensitive, and polymerase 3 is insensitive. So it is the actually scientists try to understand why this mushroom causes death. What is the basic principle behind this poison? And they found the active principle of that poison is alpha amanitin, and alpha amanitin acts differentially on these three polymerases. As they inhibit transcription to different extent in these three polymerases, that's why protein synthesis is affected. Once this mushroom or poisonous mushrooms are consumed in high dose, they cause liver dysfunction due to blockage of protein synthesis, right? And it has a long story. Anyway, but here, why I told you, because we can drive students this way. Mushroom, poisonous mushroom, there is story. Agrippina was there, Claudius Caesar, tankar story achi ta pachre. How this queen killed the king by giving this poisonous mushroom along with this edible mushroom in food, and the king was killed. Anyway, next is an alteration in the genetic material that eventually give rise to mutant progeny may originate in which of the following? An alteration in the genetic material that eventually give rise to mutant progeny may originate in which of the following? That means how mutation is created. Either the sense strand or the antisense strand of DNA. The sense strand of DNA, the antisense strand. Okay. That is in the antisense strand of DNA. Either the sense strand or the antisense strand. Okay. This one, answer would be this one. Here, relating the template and non-template, coding and non-coding, sense and antisense. Three terms are there relating the strand that is used for transcription. Clear kari kahi dauchi. Template strand is called non-coding strand, antisense strand. Jo strand transcription ho nahi, not taking part in transcription, that is called your sense strand, because mRNA ta jota theory hochi, that has the sense. So mRNA sahita G saman, excepting T and U, seita hochi sense strand, seita hochi coding strand. So you can understand it from the book well, but that is the basic fundamental behind this. But people may often they confuse why it is called sense, why it is called coding, because जो strand mRNA theory होची सेतु तो होची तेणु सेटा coding strand no, तारो जो strand रो theory होची mRNA is complementary to the template strand. So mRNA has the sense for protein, so mRNA सहित G समान सेटा हला sense strand, और template strand हला antisense strand. तो यह हबो जे जो strand रे भी there are two strands. If A is changed, definitely in another round of replication that will go lead to mutation. So either sense or strand or antisense strand, if there is a change in the genetic base sequence, they need to lead to mutation. During transcription, RNA polymerase holoenzyme. Already I have told you, RNA polymerase holoenzyme includes sigma factor and core enzyme. Okay, this binds to a gene promoter and assume a saddle-like structure. What is its DNA binding site? Tata box. Actually, in prokaryotes, there are nearly 5,000 genes. 5,000 genes. But very few RNA core enzymes. Very few RNA core enzymes. But for each gene, there is a sigma factor. There is a sigma factor. So sigma factor plus core enzyme, they recognize the total enzyme that is called holoenzyme binds to the promoter at a site called Tata box, TATA. Because this TATA sequence is -10 base sequence, -10 base sequence, it is a consensus sequence. Many genes characterized at their promoter sequence, it is found that at -10 base sequence, they carry TATA. That's why it is called Tata box. Okay. So Tata box is present at the consensus sequence. Removal of introns and joining of exons in a defined order in a transcript unit is called splicing. Right? Removal of introns, but joining back of exons is called splicing. So splicing is one aspect. Addition of capping is another aspect. Addition of poly A tail is another aspect. These three together constitute the RNA processing. RNA processing. But question comes, out of these three, which one takes first? Which one towards the end? Capping takes first, then splicing, and telling once the mRNA is released from the template. Once the mRNA is released from the template towards the 3' end, poly A, likewise 200 to 300 adenylate residues are added, and that is template independent addition. Could you follow me or not? That once mRNA is released, only adenylate residues are added, and it is template independent. It is not relating the gene sequence. It is template independent addition. So next is in an mRNA strand, the sequence of bases is this. The sequence of bases in DNA strand which served as template for synthesis of mRNA. It is its complementary sequence you will find. Okay. Which of the following sequences of mRNA will not translate completely? Yes. Because mRNA is read from 5' to 3' end. In all four options, the direction is 5' to 3'. But wherever you will find a stop codon, see where the translation will not go to completion. That means somewhere in the middle, if the stop codon is encountered, then protein synthesis or translation stops there. Here, because we have to remember what are the three stop codons. So here in the middle, there is a stop codon. Okay. Okay. So here now a stop codon is encountered in the middle of the mRNA. That's why it will not lead to completion. So thank you, and I hope you will enjoy this. Thank you. Stop कर देता
बहुत आसी लेने बस एक बात नहीं बाहर खेल की बस मोबाइल पर
Aji Botany, na?
Head of the department,
Oh,
My
you
student
see, our time has gone, so I do
just
not.
9-11, his time is over.
But ଟିକେ କହିଦଉଛି
सर
ତାପରେ
हाँ सर ଆପଣ ଆଗ କହନ୍ତୁ କି ଫାର୍ଷ୍ଟ ଅଫ ଅଲ୍ କୋଉ କୋଉ ସବଜେକ୍ଟ ଅଛି ସବୁ ମାନେ
ହଁ ଆଜି କେତେ ଜଣ ଅଛନ୍ତି?
All 30.
13 ଆମର କେତେଟା ସବଜେକ୍ଟ ଆମେ କରୁଛେ?
Including 7 subjects.
7 subjects.
Except Odia. Physics, chemistry, biology, mathematics,
हाँ.
computer science,
So
and English.
I'll tell them and I will also inform you by that, you know. So, friends, a very good morning. The director tribal scheduled tribe is, Madam Guha is here with us today. As you know, she is the secretary of your OMTES. And she has been fighting for your cause all through. Even this, training program, which is specially designed for the EMRS, teachers' capacity building in the wake of the new curriculum and new kinds of examination pattern that has been declared by CBSE. So we thought that this is very essential that we tell you with the curriculum makers, with the question setters who are coming as your resource persons to make you updated, make you abreast with information, so that it becomes beneficial for you and also beneficial for the students, in turn. I would like to inform, Secretary OMTES and Director ST that we are organizing this hand-holding training for each subject for a period of 4 days. And what we are doing, we are, going each day, let's say there are 7 consecutive days. We are taking, mathematics, physics, botany for 1 day, 1 round. Then we are going for the 2nd round. We are going for the 3rd round, and we are going for the 4th round. We have started this exercise from 17th of August, and this will continue till 28th September. I'm sure even the teachers who have attended during last, 2 days, they feel that this was necessary for them. I wish that this training program will enhance your capacity and benefit our children at large. With these words, Secretary OMTES, Director ST wanted to interact with you a little bit, and she wanted to come and she is with us. I welcome her and I would request her to speak to you.
so we'll be giving them workbooks also this year.
Workbook.
Workbook is
They will.
first year we are going to print it, I think.
This time it is
Yes,
CBSE.
yes.
Sir, there, I think they might be knowing the new exam pattern that you
That is what is being said.
At the beginning today. I have said
it will be a two-way exam. Like first it will be objective and later on either descriptive or objective depending on the board, depending on the school. So how are the teachers taking it?
it's okay, I think, because I do not know whether they are completely aware or not. Because whatever, we have a past examination.
Thank you, Dr. Rota, Director SCSTRTI, all seniors here, especially Professor Subhash Das and Professor Vijay Kumar, all other dignitaries present here, and all my teachers on the other side of the screen. I just see my assistant secretary also has joined. So first of all, I'd like to thank SCSTRTI, Dr. Rota, and the entire team, all the guests present here for this excellent initiative, because as was being told to me by Professor Subhash Das, the 12th standard paper pattern is going to actually change this year, and it was just notified some few days back. It is to the credit of SCSTRTI and all the guest faculty here that within no time they came across this very nice idea of having a interactive session, a training session for all the PGTs of our EMRS across all the schools for an entirely 1 month. So, and that too in 7 subjects excluding ODIA. So let me request all my PGTs to take advantage of this situation, because I do not know in how many states they would be even knowing that such change in exam pattern has actually come through. So I really heartily thank the entire team here for even keeping us informed about this very important paradigm shift. And I was just going through the exam pattern. The first pattern that will be somewhere around before December, that is objective type MCQ. And after some time, maybe about March, April or February, March, that time it is going to be summary based questions. And the summary based questions also are not going to be just, question answer type. When a case study based question achi, when a question which I feel are extremely important to prepare the student for competitive exams. Sir, can I just see those types of questions? Yes. So, for example, and first of all, OMR sheet. Of course, OMR sheet has been introduced some years back, even in our 10th standard. But see, when you look at OMR sheet, it is very important for the student to know from now itself how to mark the OMR sheet. Because all these things come in competitive exam. Say, Kali Jaiki Bana UPSC, OPSC, bank, Kimba Kono bhi exam re jodi bosile, ये OMR sheet रे तो सेमानंको माने exam देबो को पड़िबो एबेटारू जदि OMR sheet रे से exam देले तो obviously हमें सेमानंको तांकर future पे actually हमें orient करूछु familiarize करूछु जेटा कि I think it's an excellent step. I remember कि हमें भी जब बड़ो हेलु OMR sheet जब first time देखिलु OMR sheet माने it is so sensitive, it's computer sensitive. ସେଥିପାଇଁ ସେ ଯେ ବ୍ଲାକ୍ ଏବେ ପେନସିଲ ରୁ କରୁଛନ୍ତି ନା ପେନ୍ ରୁ କରୁଛନ୍ତି ପେନ୍ ତେବେ ପେନସିଲ ରୁ କରୁଥିଲୁ ପେନସିଲ ରୁ ଯଦି ବାହାରେ ଯଦି ଯାଏ it used to be wrong. So therefore please understand that this OMR sheet that you are actually training the students on, you are actually training them for the entire life ahead. Apart from that, the questions for March April exam, case based, situation based, open-ended question, short answer, long answer. Each of this is, I think, something literally at least net exam re net JRF re bhi emiti question asuthibo ki nahi mo jane nahi. This case based, situation based, open-ended, short answer, long answer. Each of this is very important. आपने जानिबार कथा short answer माने केते वर्ड लिखिबो, long answer माने केते वर्ड लिखिबो. Situation based जे अछि situation based I don't know माने आगामी पूर्व अभ्यास रे सेमानकर किछि भी question paper थिला कि यदि न थिला तो sir and all the seniors here will have to sit down and prepare the questions to orient the students on what can be situation based, and all are these science topics, computer science topics. So I think it's an excellent initiative by SCSTRT, all seniors here, for taking this brilliant initiative, and it's totally to the support of OMTES that this initiative has been undertaken. I'll request Jayashree to be there in as many sessions as she can, and to avail this opportunity so that we can also see to it that when exams come, this this should be an important record. This should be one important record, and PGTs, if they have any doubt, we can, with request from to sir, if required, we can have a second round also whenever required. So because it's the first time that this kind of exam is come along. At the same time, of course, that sir was just mentioning 12th standard teachers are very important. PGTs, after all postgraduate teachers. So recruitment rules are coming along, sir, by ministry. In the meantime, also we thought it was important that at least those who were serving us for the last 20 years or so, we also took a small step. Government has taken a small step in just helping them with their remuneration. So I am sure with this small little incentive that we could do in response to whatever they have been doing for so many years, I am sure that they will take the training very, very seriously. And as sir was mentioning, he will also come up with laboratory sessions, online laboratory sessions, which further will help our students and our teachers. I am sure we will get very good marks this year from our students. So my papa always says कि बृक्ष तुम्हार नाम की फले परिचय, बृक्ष तुम्हार नाम की फले परिचय बंगाली में है, मैं उड़िया में बोल देती हूँ. बृक्ष तो तोर नाम कौन हो सेटा तोर फल रे परिचय हबो. तो सेति पाई आमे एबे जते भी परिश्रम करूछु सेटा आपनोर ऊपर रहिला कि फल कौन देबे, ताहले जाय कि हमें जानिबो कि हमरो जे एते परिश्रम होजे सेटा साब्यस्त हैला. एटा कोई कि मु पुनी थोरे आपनो समस्त को बहुत बहुत धन्यवाद जनाउछि for coming in full attendance. Sir, I will please request that attendance is taken because and please share it with us at home test the attendance record, and we will see to it that all of them attend the sessions in full capacity according to their timetable. So with this, thank you, sir. Thank you all the seniors here. And thank you, teachers and UMTES. Thank you.
We have just recorded video recording.
Thank you, ma'am. या भितर आगुरु के थिले आगुरु जो क्लासेस थिला
Dear colleagues, now we, after the speech by Director STCOM, your Secretary Umtes, now we are moving for the second session. And on my left is my classmate, Professor K. Vijay Kumar, and he has retired as a DZ and Chairman, Government of India, as in the textbook publication wing of the Ministry of HRD, Government of India. Now, I have requested my friend, I have taken the liberty to take advantage of my friend, Professor K. Vijay Kumar, to address you so far as your post to syllabi and a new pattern of examination is concerned. I don't want to become a bar between him and you people. So now I hand over the mic to my classmate and friend, Professor K. Vijay Kumar, to start his deliberation. Professor K. Vijay Kumar, please.
on ଅଛି ତ ନା ନା এটা চেঞ্ଜ କରିବା ପାଇଁ ଟିକେ ପଟକୁ ବସିବାକୁ ପଡିବ ନା এটা চেঞ্ଜ କରିବା ପାଇଁ ନା ନା এটা ତ এইটি କରିଦେଲେ ହବ ତ ଆପଣ ସେୟାର କରିବେ ନାଇଁ ମୁଁ ଟିକେ 2 ମିନିଟ୍ କହିଦିଏ ତାପରେ Good morning everyone. It is still good morning, I suppose. 11:25.
Good morning, sir.
Good morning. Now, it is a wonderful experience to be with the PGTs. Now, you are, I think, attending your second class. Yes or no?
No, sir, first class.
First class. The previous
Second
class.
period.
Second period. Okay, whatever it is. The botany, you are attending the second period. Acha, fine. Now, the thing is, you are all PGTs, and you tell me, what is the difficulty in framing and answering multiple choice questions? Because I have got another 2 periods later on 1st of September. If you tell me your difficulties, I will try to adjust at that time, deal with that time. Tamara kona difficulty hochi? I hope all of you are Oriya or any
Yes, sir.
other language.
Yes, sir.
You are all Oriya. Okay, fine. Now, whatever it is, better to speak in English and also Oriya combined together. Now, tell me, what is the difficulty that you face in framing multiple choice questions, and what is the difficulty in answering them? Anyone? Quick. No one wants to answer, or no one is there?
So much choices are there. To put
Choices are there.
so any student that should be it is very difficult to choose the correct answer from out of these 4.
Okay. You have told it, but in a different way. No, usually the choices are misleading. They give such an answer which each answer appears to be correct. So
Yes,
even if you know the answer, you get confused. Is it true or not?
Yes, sir.
If you know the exact correct answer, it is okay. Otherwise, you will get confused. Now, I was looking into the CBSE instructions. I found that the questions for multiple choice should be based on assertion and reasoning. Have any one of you went into this assertion or reasoning upre base karibo? Mane kon? Ta mane ta kon?
assertion মানে
assertion
ওটা
and reasoning. গোটায় assertion মানে
स्टेटमेंट মানে false exclamation भी दिया जाता है जोटा कि स्टूडेंट को चारटा ऑप्शन मिलछि से से बाछिबो जे both assertion and reason are correct or both assertion and reason are false or assertion is the true and explanation or reason is the not proper explanation of or assertion is correct and reason is false. These four options are found.
I could not get it, but still then. Anyone else who can tell me what is assertion and reasoning? Avinash, Gyanaranjan.
Yes, sir. Student have to think logically, sir. They will be able to think logically and analytically. They have to go through the question and select their answers on the MCQs.
Acha.
Yes,
I
sir.
will tell you what does assertion and reasoning mean. Assertion and reasoning mean.
Yes, sir.
Now you have gone through the question. You said you will apply your brain and analytically analyze it and get the answer. Actually, the question is, once you think that this could be the answer, that is statement of fact. That this is the answer. That comes to your mind. This is the answer. That is assertion. Assertion is statement of fact. Now, what is the genotype of a pure tile pond? So it is capital T, capital T. That is assertion. And what is reasoning? Now, the assertion must be supported by reasoning so that your assertion is examined and found whether it is correct or wrong. The reasoning must justify your assertion.
Yes, sir.
Okay? Anyone
Yes.
has got any doubt?
No, sir.
You have a doubt? No.
No, sir.
Second type of questions that CBSE suggests is the case-based. Now, case-based is most important as far as this genetics is concerned and many other subjects are concerned. Case-based means, suppose we say, if this type of plant is crossed with this type of plant, what is the type of offspring you are going to get? That is the case. A tall plant crossed with dwarf plant, that is both are heterozygous, then one is heterozygous, one is homozygous, then what will be the progeny? That is the case-based. So two types of questions you have to frame and the students have to answer, but mostly the questions we get in examination are direct to the point, many questions. So we will deal with all such type of questions. Today, I was not knowing your mind, so I have prepared some questions on my PowerPoint. Each slide on the PowerPoint has 3 questions, and we will just discuss them one after the other. 3, then 3, then 3. Like this, we will go on as far as we can. Now, this mostly includes heredity, and I have kept for the next class, other class, the rest of the portions that are there. Now, here I want to tell you one thing, that because it is in this state, we have separated botany and zoology. The teachers are different, but it is expected that both the things should be taught together because most of the things are linked. Now, whatever it is, we will try to deal with this thing to the best of our abilities. Let us see what we can do. Now, I will ask my friend to. That is a problem. Now there is a problem in showing the PowerPoint. Now, so we have discussed what is the, acha, here one more thing that you may face in the class about MCQs. Now students are mostly interested about, suppose you are teaching genetics. So your portion is something the student may ask, what is Klinefelter syndrome? What is Down syndrome? This all comes in zoology. I totally agree. But you must have a brief idea of it so that you will not have a poor thing. Don't say that you ask your zoology teachers, give some idea and then ask the student to go to the zoology teacher for details. So you have to have a brief idea. It is there in your council book, and it is also there in CBSE NCERT textbook and other books that support them. And you can very well study them and get your answers. Now there is some problem in this PowerPoint. I'll share. Once it is shared, I will get back to you. Now, Acha. Okay. Now, one thing I may make it very clear. These PowerPoint slides are prepared by myself. So there may be some spelling errors. It is a typographical mistake. And I am not as computer savvy as you people are, as we are old generation, but still then I have tried. Now, you will find one thing, that the questions are not numbered, because once I started numbering the questions, I found that it is moving this side, that side, and getting disturbed. So I have made the page numbers. Now, from page number, I will know which question you are asking me about. Now let us see the questions. Now, one after the, I have put here 3 questions. Now, these 3 questions, you try to answer them, and after that, if we will discuss it. And as I told you, 2 things. First is assertion, second is reasoning. Now, when you are answering, please switch on your mic. No, thank for anyone? No answer.
First is all of these. First is all of these outbreeding devices help in increasing the hybrid vigor, increases the adaptation, and reduces the expression of lethal genes. All are correct. All are correct.
This is your assertion. What is the explanation or reasoning?
Because outbreeding devices are followed by the plant to increase to adapt the cross pollination and to prevent the self pollination, because self pollination
outbreeding is cross pollination. Cross pollination is outbreeding.
Yes, sir.
Okay.
Yes.
So, okay. So you say that all correct. Anyone who differs from that answer? Anyone who counters that answer? No. No one is countering the answer. Someone has, I think.
Sir, when two unrelated people have a child, that is the best example of outbreeding.
What? Two unrelated people
Have a child.
That is?
It is an example
Hybrid
of outbreeding, sir. Best. Sir.
That is okay. But whether all correct is correct answer or wrong answer?
All correct is the correct answer, sir.
Right. All correct is the correct answer. I have got two correct answers. So I agree with what you say. Hybrid vigor body wo. Mane outbreeding ko ame hybridization koi pariya? Yes or no?
Yes, sir.
Yes.
Yes, sir.
That's okay.
Yes, sir.
Yes, we can ask it outbreeding. Now, these things you must remember, because in place of outbreeding, you may write hybridization. Hybridization increases hybrid vigor. Now, increased adaptation, because a number of genes are coming together, reduces expression of lethal genes, because if a lethal gene is present, that is suppressed by the normal gene, which could be present in the other partner. So that's why the all correct is the right answer. Very good. Now, heredity lies within
Species.
reasoning.
Yes, sir.
Someone said genus.
Because
okay. Because
in between two similar species, the interbreeding should be possible, and the genetical characteristics should be transmitted from one generation to next generation. So species, in between a species, your heredity should be found.
Acha, your reasoning is right, but you are explaining it in different way. Okay, fine. It is very good. Now, someone said genus. Someone said genus. Give your reasoning.
Hello.
Nine.
Yes, sir. I think it is genus. The heredity. Hello.
Heredity.
As the genes are being of the offspring inherits from the parent, so I think it is from the genus. Heredity has a it is from genus. That is it from the parents to the offspring. So it is the genetic material.
Now answer that.
Sir, genus.
Genus is
Yes,
fine.
sir.
You see, this is the ambiguity caused by multiple questions. One side it is seeds, and the other side it is genus. Okay. Anyone else who want to say that it is class? Anyone else who want to say that it is kingdom? None. So I will give the reasoning. You tell me the answer. What is heredity? Heredity is transmission of characters from parents to the offsprings and from generation to generation. Now tell me, I have given you one statement. The other statement is one reason. The other statement is a brinjal plant gives rise to brinjal plant. And what is the botanical name of brinjal plant?
Solanum
melangana. Any other species of solanum?
Solanum tuberosum.
Solanum tuberosum. Now tell me whether the genes sprouts from Solanum tuberosum to Solanum melongana?
No, sir, because
Solanum melongana. No, let me ask. Solanum tuberosum to Solanum melongana, Solanum melongana to Solanum tuberosum, or it process from Solanum melongana to Solanum melongana, Solanum tuberosum to Solanum tuberosum. Now tell me. I will tell you the answer. It passed from Solanum melongana to Solanum melongana, and it is a species. It is not a genus. If you say it is a genus, then genes must pass from Solanum melongana to Solanum tuberosum and Solanum tuberosum to Solanum melongana, because both belong to the same genus. So it is not within the genus, it is within the species. Anyone who has doubt, please ask me. So the first answer which was given is correct. It doesn't pass from genus to genus, it is within the species. Now we may cross different species, that is a different issue. But as the first lady said, we cross between the varieties of the same species, not different varieties. Because P has many contrasting characters, so we crossed Pisum sativum with Pisum sativum, not Pisum sativum something else. So it is within this species. Now, I hope the answer is clear. Next, segregation of genes occurs in, the 3 is wrong there. Tell me. Anaphase 1. Here, this is a straight answer. There is no much of ambiguity. When in meiosis 1, meiosis 1 is the anaphase 1 is the correct answer. Meiosis 1, the synapsis takes place in prophase and crossing over takes place. And after crossing over, segregation of the chromosome takes place in anaphase 1. That is why it is, the correct answer is anaphase 1. Certain things I am moving fast because I have made around 140 questions. We cannot cover, but whatever it is. Now, anaphase 1. The main purpose is to give you an idea as to how to deal with these questions. So, anaphase 1. Any doubt?
No, sir.
Anaphase, no doubt. Anyone else?
No, sir. No.
No. Okay. To see the effect of sex on inheritance, Mendel performed
Back cross. Back cross भी है और test cross भी हो. Both है.
back cross above, test cross above. Anyone else?
Because test cross is the another way of the back cross.
Okay. Anyone else?
Both answers are right, sir. Test cross and back cross.
Both are right. Anyone else?
Test cross and back cross.
Which of the following statement is incorrect?
Acha, ebe subila. Are you able to listen?
No sir.
Shuna jauchi? Ha, subuchi?
ଏବେ ଶୁଭିଲା sir, ଶୁଭୁ ନଥିଲା।
Acha, ठीक ଅଛି. Now two PGTs have answered the question. Now both of you are wrong. Now both of you are wrong. The reason being, now you see, now the first three questions were answered by Sangeeta, I think I had her name. Now she answered them very correctly. But when it came to fourth question, she made a mistake. Now that happens when we stumble that way. And in the second questions, one or two people said it is genus, not species. So when we are stumbling, think about the student. Now I will also stumble. I also cannot answer all questions. Now, so what is the cross? Now, the correct answer is
Reciprocal
before
cross.
giving the reciprocal cross. What is reciprocal?
No, sir, it's
Exchange.
actually outcross, I think.
Male, making male female and making female male. That is reciprocal. So to effect of sex, mane tall ta male, dwarf ta female. प्रथमे कले तापरे dwarf ta male, tall ta female कले कौन भावची? So reciprocal, that is reciprocal cross. It is neither test cross nor back cross. Now, as you were tempted, a student will also be tempted that way. ताको भी लगियो, अच्छा test cross था तो test पाई हुए, एटा निश्चय है थियो. So that misleading thing is quite possible. Now, so the correct answer is reciprocal cross. If you have any doubt, any question, stop me there itself. Now, which of the following statement is correct?
Incorrect.
I'm sorry, incorrect. कौन से कोई statement था बोल? Which of the following statements is incorrect?
None of the above.
None of the above. Anyone else? ताले किसी भूल नहीं बोली तो मैं कहूँगा.
Yes sir.
How किए? Anyone else?
Others please interact.
Others are not interacting. Please interact. Unless you interact, how can you know?
Other PGTs, please interact.
There is no purpose of this.
If you don't interact and do this. Because your director is very serious regarding this. And there is a hidden proposal that it may be related to your promotion in future. Non-participation means you can imagine what to be think. Those who are participating, much help will be given to them.
If you have doubt, ask. We are not able to get it, please explain. I will explain it, but don't keep silent.
Other PGTs, please act.
Sir, it should be none of the above.
None of the above. Achha. Very good. That is the correct answer. Now, here in this particular question, I will tell you what is the problem. When I read the question, in what way I read, which of the following statement is correct? Intentionally, I did it because a student or a teacher normally makes that mistake. Kota correct? Prathamta dikhila, gene is sequence of nucleotides. Tick kar diyo. Eta hochi correct. Sarla, that is the answer. So that way you make a mistake and students make a mistake. Ask students to repeatedly read the question and then answer it, not simply answering it just like that. don't be in a hurry. And not correct, incorrect, these are very misleading words which will put you in trouble. Acha. Next. At least I must get 3 answers for this. Drosophila of plant kingdom is
P, sir.
P.
Green mold. Green mold.
Green mold. Acha. P, green mold. Then आगुटे किए आगुटे नमिता रथ कहले जे इट इज ग्रीन मोड आउ जण कहले 3 जण कहले रंजन व्हाट इज योर आंसर
Pink bread mold.
प्रिंट ब्रेड मोड अच्छा अलगा अलगा ग्रीन मोड पी पिंक ब्रेड मोड आउ जण मेज कह दियो और चॉइसेस सब anyone else? This is the beauty of multiple choice questions, and the examiner always tries to put you in trouble.
Bread mold.
Avinash?
Bread mold.
Bread mold. Acha, pink bread mold. Acha. So I will give you the answer now. It is A, pink bread mold. 2 of you, last 2 are correct. First 2 are wrong. Now, pink bread mold is Neurospora crassa. It is the Drosophila of plant kingdom. Now, Neurospora. I did not intentionally mention Neurospora. Neurospora re केमिति क्रॉसिंग ओवर होची कोन केमिति स्पोर डिस्ट्रीब्यूशन होची पूरा क्लियरली देखा जीवो बिकॉज ऑफ द डिफरेंट कलर्स ऑफ स्पोर्स इन द पॉड्स दैट इज 8 spores 8 spores so pink bread mold Neurospora okay acha Mendel conducted his hybridization experiments with garden pea for केते वर्ष नो नो नहीं ये टिके को की चाहिए ए हाँ लेमन टी हाँ को
7 years.
7 के कहला एटा
Or 7 years, sir. Right answer.
7 आउ की आंसर अछि
7 years.
7 की संगीता कहू 7 ना नहीं
8 years, sir.
7 years, sir.
7 years.
हाँ
Excuse me जो मैडम को पाखे छुआ छी टिके काम काकू छुआ को टिके पाखे रखिबेनी प्लीज काने एटा रेकॉर्ड के गवर्नमेंट को जीवो
हां
जे जो रेकॉर्डिंग के चली छी गोटनो जे जा पाखे मैडम को छुआ छन टिके काइंडली
बेनजीर व्हाट यू से
सेपरेट रखी छी
बेनजीर एटा चेन्नई से
Many
okay
years.
many years
Sir,
okay
7 years, sir.
7
Right
years.
answer.
7 years so I want 7 years 8 years many years acha namita no answer okay
8 years.
the right answer is 7 years now reasoning is difficult
Sir, 1856 to 1863.
1856 through
63.
1863 केते वर्ष होला
56 to 63.
अला केते वर्ष
केते वर्ष होला हमें
7 years, sir.
7 years, sir.
7 years okay 7 years अच्छा एटा कोठी पढ़िला तमे केते भयरे देती 8 इयर्स
1856 ରୁ 1823
हां how I will give you the correct answer. 7 is the correct answer. It is right. Now I find this number in the NCERT textbooks. So those who have attended my previous classes, I have told you that as far as CBSE is concerned, the Bible for CBSE or Gita for CBSE is NCERT textbook. And somewhere I found 7 years as per NCERT. It is also written. So here the answer is 7. 7 print in your mind. Now next question is a difficult or an easy one. Tell me, next question. The question number is third page, second.
Exact question would have stated as.
What is the answer?
1 is it 1 is 2
1 1 1 1 1 2 1 0 0 3 1 0 1 1 1 0. Which one is the right answer? चिंता करूच very good. तमु एटा होची right question. This is a question which is based on case based question. Given you a case या पूर्वटा हला knowledge based question. This is case based question. Now a case रे कोन होबो? Case टा होची cross is between these two. Now what will be the ratio?
The answer is B.
Answer is B. Kiya kochi?
Bhagyasri.
Rajasri. Thank you. Why? Anyone else? Anyone else?
Sir, answer is B.
B. Thank you. Why? Why the answer is B?
Showing the quantitative inheritance, sir. Polygenetic inheritance occur.
It is not quantitative. It is qualitative. We are showing here, we are not telling about quantitative or qualitative. We are simply giving you the genotypes. Tell me, it is not quantitative. Anyone? Reasoning kon dabo. I tell you the answer, it is B is correct. But why?
Small a is absent in first A, B, B is small a absent, sir.
Small a? Acha. Okay, I will explain it. एटा देखि कि मते को ए जो क्रॉस टा करा गला सेटा की क्रॉस मोनो हाइब्रिड ना डाई हाइब्रिड ना ट्राई हाइब्रिड ना की क्रॉस
Try hybrid.
डाई हाइब्रिड काहे कि ना डेटा जीन नहीं कि हां
Yes, sir.
यस सर हाउ ये काउंटर कॉलोनी मु वर्तमान आंसर टा कहूची this is a mono hybrid cross. Thank you. She is astonished. Why? How can it be mono hybrid? सुनो तमे 1:1 रेशियो जेते बेले कहूचो सेटा डाई हाइब्रिड हबो केमते सेटा छाडो यार आंसर टा हला तमे देखो से जीनोटाइप को कैपिटल ए होमोजाइगस ना हेटेरोजाइगस
Homozygous.
होमोजाइगस स्मॉल ए होमोजाइगस ना हेटेरोजाइगस
Homozygous.
homozygous. Okay. Now, when you are crossing capital B, अच्छा, ET, दुईटा capital B, small b, capital B, small b. तमर कौन है इटा? Same ना different? Same capital B टा का planter same. Now, Mendel जेतबले गोटे मते उत्तर दियो. Mendel जेतबले tall plant नेले ताधरे अन्य छोटा कैरेक्टर थिला ना नथिला ना जानि न अन्य कैरेक्टर थिला किंतु नेले केतुटा से केतुटा नेले गोटे नै थिले एठी देखो अन्यटार किछि मान्य नै किंतु एट हाइब्रिड टा होची कैपिटल बी स्मॉल बी कोटा हाइब्रिड कोटा कैपिटल ए एंड small a capital A and small a ए दीटा रे डिफरेंस अछि ए दीटा मिसले कोन रिजल्ट बाहरिबो कैपिटल ए रे गैमेट होची ए
स्मॉल ए
आउ कोन रिजल्ट बाहरिबो इटु गैमेट कोन हबो मु कहूची कैपिटल ए कैपिटल बी एम आई ओके
Yes, sir.
कैपिटल ए
yes sir
हाँ कैपिटल ए स्मॉल बी फाइन
Yes.
sir
देन स्मॉल ए कैपिटल बी
then
स्मॉल ए
small
कैपिटल बी स्मॉल बी तांको भितरे तमे जते क्रॉस करूछो इटी हेटेरोजाइगस कंडीशन केतोटा अछि
कोन पड़ेगो
नुआ हेटेरोजाइगस कंडीशन केतरा आसला गोटिए सेइथिरु तम रेशियो आसीबो 1:1 तमर जो ऑफस्प्रिंग्स देखौची तादरे हेटेरोजाइगस कोटा अछि हेटेरोजाइगस कोटा अछि
ए कैपिटल ए स्मॉल बी स्मॉल बी
नहीं हां सेटा बोथ हेटेरोजाइगस हला आ गोटे हेटेरोजाइगस
कैपिटल ए स्मॉल ए कैपिटल बी
हां एटा गोटे हेटेरोजाइगस आ गोटे हेटेरोजाइगस हला
Capital A, small b.
स्मॉल बी तो कोटा इठी दुई हबो हाइब्रिड जोटा
कैपिटल ए
कैपिटल स्मॉल ए कैपिटल बी स्मॉल बी हला एठी सबुथिरे कैपिटल ए अछि ना नै प्रथम तीनटा रे
अच्छी सर
अछि ताले फिनोटाइपिकली समस्तंकर फिनोटाइप कोन हबो कैपिटल ए ही हबो से प्रथम तीनटा लास्ट छाड़ दियो हां तारो फिनोटाइप हबो कैपिटल ए ना नहीं
यस सर
फिनोटाइप अलगा किंतु फिनोटाइप कोन हबो कैपिटल ए यार फिनोटाइप ता कैपिटल ए हबो ना नहीं बी कथा छाड़ो वर्तमान यार फिनोटाइप कैपिटल ए हबो केतरा कैपिटल ए प्रथमटा एक द्वितीयटा दुई हाइब्रिड तृतीयटा एक ताले हमर केतरा 1:2:1 1:2:1 I mean capital A small a 1:2:1 in the above. Now again tell me whether it is a monohybrid or dihybrid cross, whether I am right or you were right. Now think and tell me. Now I will be putting you in soup a number of times. You have to be firm on your assertion and reasoning. You must be firm. Mu jaha कहली सेटा को मानि जीवन अच्छा how खाली क्लू दउची मते परे कहबो तमे अच्छा ए जो क्रॉस टारे तमरो ए डायहाइब्रिड क्रॉस रो रेशियो केते डायहाइब्रिड क्रॉस रो रेशियो केते
I don't know why you, the PGTs, are silent this thing. Sir is asking what is the ratio of dihybrid cross. This is not a thing to be remembered. But none of you are here. No, I mean to say,
9:3:1.
why don't you answer that way
why don't you answer? It is an interaction session. I have to take multiple choice type of question, and I find except one madam, none of you are answering and taking any action. Now, what is the necessity of conducting this type of orientation program if you don't participate? We are not inviting you to sit before the camera. Please, you take it pretty seriously, this thing. Okay.
Okay. On पहले अच्छा 9331 अच्छा मते बहुत फास्ट वाला डायहाइब्रिड क्रॉस रो हां हां 9:3:3:1 now जीनोटाइपिक रेशियो कहते
1:2:2:2 something
what is the genotypic ratio of dihybrid cross
1:2:4
1:2:4 और पढ़ोगे मुझे कौन रेशियो के कोई परियों
1:2:1:2:4:2:1:1
You go either in ascending series or in descending series.
ना ठीक अच्छी से जहाँ कहूँचंती may be correct or may be wrong. I will give you one simple procedure to remember it. The ratio is 1:1, 2:4:2, 1:1. 1:2:4:2, 1:1. Is it right or wrong? 1:1, 2:4:2,
1:2:4:1:2:1:2:1.
Okay. Acha, that is not the easy way to remember. Now, if you have pen and paper, write there. Practically 1:1. लिख लो तुम कागज रे लिखो 1:1 15 हला
1:1
Please, you don't. Program seriously, and you are talking amongst yourself, and neither you are replying to the research persons. Now, if you don't amend yourself, I would like to stop your training program. Jodi sincerely next program botany stop. I will stop the program in botany completely. I'm very much satisfied with your behavior and performance. सर सब म्यूट कर दे तमे किछि तमे कर के चलो किछि बच्चे लाभ हो किछि सेमी कर
आपण लेट जाय ने आपण जा से कहले बाकी सब लेट
दरमा दरकार
सर 1:2:2:4:1:2:1:2:1.
Okay.
So don't interest.
How ତମେ ଯଦି ଇଣ୍ଟରାକ୍ଟ କରିବନି ଆକୁ କଣ୍ଟିନ୍ୟୁ କରିବାର କିଛି ନାହିଁ ଖାଲି ମୁଁ ଆନ୍ସର ଟା କହିଦେବି
सर टिके तार इजी प्रोसेस टा टिके कोंतु सर
हां हां ସେଇଟା दैट
सर इजी मेथड टा टिके
ମୁଁ କହିବି ନା 1 to 1 ଲେଖିଲ ତମକୁ କହିଥିଲି
हां सर
यस सर 1:2:1 यस या
अच्छा 1 सामनारे तीनटा गार टाण एमिति मु देखा जाउची तांको तो हां एमिति फोर्थ लाइन भलिया 1 2 3 2 पाखरे तीनटा लाइन टाण आउ थरे 1 पाखरे तीनटा लाइन टाण एठी मु देखाइ दउची तुम जदि मु देखा न ब्लैक बोर्ड आसो ना ना एठी नाउ एठी 1 अछि 1 कैपिटल T ता सामना रे एम्प्टी तीनटा गार तापरे 2 सामना रे एम्प्टी तापरे 1 एमिटी हला 1 to 1 सामना रे तीनटा सेठी आउ थरे 1 to 1 2 4 2 लिख दियो लिखिलो 11 टाइम लगी
हां सर
तो कौन लिखि सर
एठी गोटे कहि दोची देखो 1 लिखिलो ता सामना रे एम्ति तीनटा 11 टाणिलो इठी 1 to 1 लिखो ता सामना रे 1 to 1
हां सर
तापरे 2 लिखिचो ता सामना रे तीनटा 11 1 to 1 हला
यस सर
हां तापरे 1 अछि ता सामना रे तीनटा 11 1 to
करे करिवो नहीं चलती करे करिवो
गुणी दियो प्रथम टाकू द्वितीयटा रे गुणी दियो हां एमती लिखि दलो ना देखा जाउनी देखा जाउची
जଣା पडुनी
देखा जाउनी सेटा अच्छा हेलो हाउ ठीक अछि हमर गुणिले केते आछला ए जो 1 to 1 अछि याको या सहित गुण मल्टीप्लाई करो एक रे एक मल्टीप्लाई माने एक सहित एक केते आछला एक दुई सहित एक केते आछला दुई एक सहित दुई एठी आछिबो आंसर देखो 1 2 1 2 4 2 1 2 2 1 1 2 1 2 4 2 1 2 1 बुझियला ना नहीं
यस सर
यस सर
अच्छा इट इज जति तमे फिनोटाइप रो करछो फिनोटाइप रो रेशियो केते
9:3:1 3:1
3 सामने एटा गार टाणो सेटा दीटा अछि त तीनटा थिला 3 सामने रे दीटा गार एंती टाणो 1 सामने रे दीटा गार टाणो 1 सामने रे दीटा 3 सामने रे दीटा सेठी पुनी 3 1 3 1 लिख दियो 1 सामने रे दीटा गार गोटे 4 आउ एक सामने रे दीटा गार गोटे 4 3 1 अच्छा
यस सर
केते आसेला गुण
9:3:1
बारला आसेला की
Yes, sir.
तापरे 9:3:1 आसेला अच्छा से 9331 को बोली जाओ 3 सामने रे आउ थरे दीदीटा 4 कलो
हैं
एको 1 3 1 लिखलो 3 1 लिखलो
सुबुनी
ताको गुणिलो 3 1 3 1 सहित तापरे 3 1 3 1 जो रहला चारिटा ताको आउट रे 3 1 3 1 3 1 3 1 रे गुण चारटा केते आछि
सर बुझी हेलानी आथरे टिके बुझान
बुझी हेलानी अच्छा सुनो एठी सेठी तो देखा जाउनी तुमको एठी 3 1 लिखलो ये को ना बुझा करूची 3 1 याकू गुटे फोर्थ लाइन देलो इठी गुटे फोर्थ देलो 3 1 3 1 एटा गुणिले केते आसला सेटा छाड़ दियो एठी 3 1 एठी 3 1 आसला तापरे याकू पुणी एटा 2 एटा 2 एटा 2 एटा 2 3 1 3 1 3 1 3 1 हला बे
यस सर
हां एबे से तीनटा जाकू गुण 3 into 3 into 3 केते आसला 3 into 3 into 3 केते आसला
3 to 3 9 9
into 3 तीनटा अछि ना
27
मूल को जाओ केते आछि
27
हां हां
27
हां ता तळ को केते आसीबो 27 तळे 9 ता तळे 9 ता तळे
3
9
हां ता तळे
3
हां तापरे
9
3 9 9 आसीबो नियो
3 आसीबो 3
3 3
हां
सर
3 1 आसीबो हला केते आसीला रेशियो
9 is 27
27 is to 9 is to 9 is to 9 is to 3
is to 3 is to 3 is to 1 this is tri-hybrid phenotypic ratio hela
yes sir
this is tri-hybrid phenotypic ratio याको तमे चेकर बोर्ड करके करिया दरकार नहीं नाउ जीनोटाइपिक रेशियो केमती आसीबो तुमको कोन कहली 1 to 1 2 4 to 1 to 1 ताकू पुणी 1 to 1 2 4 to 1 to 1 1 to 1 2 4 to 1 to 1 केमती गुणी दियो 1 to 1 रे सब गुडा गुणी दियो जहा आसीबो उटा हला जीनोटाइपिक रेशियो तमे गेस भी कर परियो 1 जोठी रे रहला नानी जीनोटाइपिक रेशियो केते मते कहलो ए डाइहाइब्रिड रो 1 to 1 2 4 to 1 to 1 1 को पुणी 1 to 1 रे गुणले केते आसीबो
1 to 1
पुणी 1 to 1 1 to 1 रे सब को गुण सब जाक माने तीनटा पुणी तीनटा दैट इज ट्राइहाइब्रिड रेशियो मत पुणी के कहलो जल्दी करो
कौन कहले
तुमको डायबिटीज अछि ना
1 to 1 2 4 2 1 to 1
हां एतकी पूराटा 1 to 1 2 4 to 1 to 1 तो डाइहाइब्रिड हे गला ताको पुणी 1 to 1 रे प्रत्येकटा को गुण केते आछि प्रत्येकटा माने पूरा मूल जाय मु भाबिली मु 140 टा क्वेश्चन करछि मु खाली ओ देखो पट्टी ओदे अधिका दरकार पड़ियो याको देखले दरकार ही ना कौन बाहर कर पारलो
No, sir.
हाँ को
जानी पारना sir. जानी पारनो sir. जानी होनी.
जानी पारलो अच्छा
ना.
ए बोर्ड ता देखा जाउची की
हाँ हाँ बोलो
ना
Yes,
देखा
sir.
जाउनी पूरा तो
अधा देखा
बोर्ड ता
जाउची.
देखा जाउची
अधा देखा जाउची.
सर व्हाइट बोर्ड टा उठी पाखु काले देखा जबो
मेरा तो पाखु नहीं है एबे दिसला
हाँ sir.
एबे दिसची तो
हाँ sir.
लाइट टा लिबे चले दिसयो अच्छा इटा डायहाइब्रिड क्रॉप जानि परला तो जीनोटाइपिक रेशियो 1 to 1 2 4 to 1 to 1 एबे याकू आउ थरे ना ना एटी देखिलो कौन कली मु सबुटी पुणी तीन भाग एटा को कली everywhere again trifurcated now 80 मध्य again 1 to 1 1 to 1 1 to 1 1 to 1 like this गुणिबो केमती 1 into 1 into 1 into 1 केते आसीबो
1.
1 तापरे 2 into 1 into 1 into 1
2.
एटा केते 2
2.
एटा
1.
1 एटा
1 2
2.
2 हाँ 2 एटा
क्या डिफरेंस 4 1 1
हाँ
1 2 2 जा 4 4 2 जा 8 8 2 जा 16 8 1 जा 8
2 ना एटा
दुई दुई 4
एक दुई दुई एक कौन आसो
4
और
हाँ 4 ना
4
1 into 2 into 2 into 1 केते
4
4 एटा केते आसो 2 into 2 into 2
8
8 एटा 1 into 2 into 2
4
4 एटा नेक्स्ट केते आसो जल्दी को एटा केते आसो एटा जा एटा से प्रथम जा एटा से को
only 1 to 1 আছে
1 to 1 very good it is again 1 to 1 how 80 देखो
then again
1
2
to एटा 2 1 into 2 into 1 into 2 केते
4 2 আছে
4 4 आसी एटा
2 2
4 एटा 2 into 2 into 2 केते
8 8
8 एटा 1 into 2 into 2 4 एटा केते आसीबो एटा को नहीं 4 into 2 into 2 केते
16
16 4 into 2 into 2 16 एटा 2 into 4 into 2 into 2 केते 8 दूने 16 दूने 32 is it right Is it right?
32, 32, sir.
32. Eta? 4 दूने 8 दूने 16.
16.
Eta? Eta कितने? 242 ना नहीं? Eta 242 तो?
हाँ
Eta 242?
में 8
এটা 2 2 into 2 2 2 2 4 2 না কত এটা
8, sir, 8.
না না এটা আসবো 4 8 চেক কৰিব 1
रबर कुछ पाइले रबर दिया है ना
कौन बैंडविड्थ चेंज हो जाउची पळै जाउची सबले गोड़ बदलुଛନ୍ତି ସେ ତାର ଫଳରେ ସିଷ୍ଟମ ଚେଞ୍ଜ ହେଉଛି ତାର କାଇଁ ଗୋड़ ବଦଳୁଛନ୍ତି ଆମ ସେପଟେ ବସୁଛନ୍ତି ତାକୁ ମଇଠି ମୋଟା କରିକି ଥୋଇଛି ତାଙ୍କ ଅବସ୍ଥା ଅଟୋମେଟିକ ବଡି ଗାଡି ପଳଉଛି ମ୍ୟୁଜିକ ଯାଉଛି
अच्छा सुनो ସେଇଠି ଗୋଟେ ଭୁଲ ଅଛି ତମେ ତାକୁ ଟିକେ କରେକ୍ସନ କର ଠିକ ଆସିଯିବ ଦେଖ ମୁଁ এটা ଦେଖେଇ ପାରିବି ଯଦି ମୁଁ କହନ୍ତି
फुल स्क्रीन कर देबे कि व्हाइट बोर्ड का व्हाइट बोर्ड का फुल स्क्रीन कर दो
ना ना ଏବେ ଦରକାର
शेयरिंग काट दो ओटा दिसुची व्हाइट बोर्ड पे
व्हाइट बोर्ड देखा जाउची
Not clearly, sir. Clearly देखा जा
अच्छा नॉट क्लियर ताले व्हाइट बोर्ड टा व्हाइट बोर्ड टा को स्क्रीन को ले जाओ
सर शेयर स्क्रीन टा के बंद कर दो
बंद करके तापर आउतरे फोन कर कोटा एटा ठीक अछि तो
आगे
Now you multiply and tell me what is the outcome. ଏବେ ଦେଖାଯାଉଛି? Is it visible?
Yes, sir.
Yes, sir.
ଏବେ କଥାରେ ଭୁଲ ହେଇଯାଇଥିଲା ସେଠି କଣ ଭୁଲ ହେଇଥିଲା ମୁଁ କହିବି ଏଟା ମତେ ମଲ୍ଟିପ୍ଲାଇ କରିକି କହିଲ
1, 1, 1, 1.
କେତେ ନମ୍ବର୍ସ ଆସୁଛି କେତେ ଆସୁଛି ଆଉ ଏଇନି
Sir, ଆପଣ କରିଦିଅନ୍ତୁ।
ଆଚ୍ଛା ହଉ 1 2 1 କେତେ ଆସିବ
1, 2, 1, 2.
ଆଚ୍ଛା 1 1 1 ए 1 1 1 କେତେ
1
1,
1
2 1 1 କେତେ
2,
2 ଏଇଟା ତମେ କହି ଯାଅ
1, 1,
1,
କୋଉଟା
2,
4
2,
2 ଏଇଟା
4,
ଏଇଟା
4
2,
sorry
2
1, sir. 2,
हां एटा 1 नेक्स्ट
4, 2,
हां 2 एटा
1,
1 एटा एटा केते
2,
2 हां एटा
4, 4,
4
2,
एटा
2,
2 एटा
4
4 नेक्स्ट
8
एटा
2
हां
2
4,
4
4 हां एटा
2
2
2
एटा
2 4 2
एटा केते
4
एटा 2 एटा केते हो 2 into 1 into 2 केते
4
4 एटा 1 into 1 into 2
2
2,
1 into 2 1 into 1 into 1 1
1
1,
2 1 into
1 2
9 2 into 1 into 1 2
2
1 into 1 into 1
1
1.
1 into 1 into 1 1 then 2 into 1 into 1 2 1 into 1 into 1 1 तापरे इटा केते आसलो
2 4 2
1 into 2 into 1 2 2 into 2 into 1 केते
4
4,
4 आउ इटा हला
2
1 इटी तीनटा
तापर 1 2
हां
तापर 1 2
Yes.
1 लास्ट
हां एबे वर्तमान इटी कोन ओटे भूल हला अच्छा मते कुआ वर्तमान तमरो आ गुट्टे रही गला मते एटार रही गला एटा 2 आसीबो एटा रही गला 1 रो मु आ करि परनी एई जो 1 रो
Yes.
अच्छा
1 2 1 सर लास्ट ग्रुप
केते आसीबो
1 to
1
1,
2 1 सर
1 to 1 आसला वर्तमान
हाँ,
रेशियो
सर।
टा कहले ट्राई हाइब्रिड जीनोटाइपिक रेशियो
हाइब्रिड जीनोटाइपिक रेशियो
ट्राई हाइब्रिड जीनोटाइपिक रेशियो अच्छा ता पूर्वर आउ थरे को डाई हाइब्रिड जीनोटाइपिक रेशियो केते मु जो कहथि
1:2:4:2
आउ थरे
2:1:2
सुनो सिंपल वे होची 1 to 1 2 4 to 1 to 1 1 to 1 2 4 to 1 to 1 बुझेला ना नहीं
1 to 1
Yes,
to
sir.
1 to 1
Sir, बुझेला
1 to 1 2 4 to
1 to 1 2 4 2 1 to 1
1 to 1 2 4 to 1 to 1 this is dihybrid cross
yes sir
एबे बुझेला ना नहीं
Yes,
1
sir.
to 1
हाँ हाँ
1 to 1 अच्छा एठी लास्ट को देखो ए जो एकटा अछि एटा केते आसीबो ए जो एकर केते आसीबो मझिटा पर देखिबा एठी केते आसीबो एकटा मु लेखिनी
1 to 1
2
2 4 2 1 to 1
1 to 1 2 4 to 1 to 1 ताले ऊपर रहला 1 to 1 2 4 to 1 to 1 तळे रहला 1 to 1 2 4 to 1 to 1
1 to 1
एठी केते
1 to 1
आसीबो
2 4 2 4 2 8 4 2 4 2
बुझेला
Yes, sir.
again
it is ratio केते 1 to 1 2 4 to 1 to 1 then 2 4 2 4 8 4 1 to 2 4 2 1 to 1 2 4 2 1 to 1 this is what tri hybrid genotypic ratio तमे चेक करबा दरकार नहीं किछि दरकार नहीं टेट्रा हाइब्रिड दरकार माने आउ थरे ताको 1 to 1 रे गुण दो एक मन रखता simple answer is the genotype starts with the 1 to 1 and ends with 1 to 1 देखो एठी 1 to 1 एठी 1 to 1 तापरे केते आसबो 2 4 2 तापरे 1 to 1 तापरे टेट्रा हाइब्रिड रे कोन हबो ए जो 2 4 2 4 8 4 अछि एटा 4 8 4 8 16 8 4 8 4 एमती आसी आउरी बढ़ जिवो टेट्रा हाइब्रिड पड़ते तो जीवा दरकार नहीं एंड एटा होची इजीएस्ट वे तमे किछि गणिया दरकार नहीं तमे जो गणित नंबर ता लेक्चर नो इट इज वेरी सिंपल जोटी एक आसला सेठी केते आछि जोटी एक आसला
एक
ना नहीं जोटी एक आसला 1 इट इज 1 to 1
Three,
बुझेला
three.
ना एक आसले इट इज 1 to 1 दुई आसले
2 4 2
2
2
4
4 2
कोणी एक केते
1 to 1
1 to 1
1 to 1.
माने
2 4 2
ना एटा दुई आसला परा दुई दुने 4 दुई आसला माने 4
2 4 2
हां बुझिलो तो दुटी गुणन आसला सेठी बेसी आसीबो 2 into 2 बढ़ी जीवो सेटा माने जोटी तमरो जेतिके आछि ता अनुसार तार वैल्यू टा बढ़ी जीवो किंतु गोटे सिंपल टेक्निक मन रखो जेतिके तमरो आछि टेट्रा हाइब्रिड तो केबे भी आसबनी तमरो कंपटीशन रे 1 to 1 2 4 2 1 to 1 is the magic number 1 to 1 2 4 2 1 to 1 ट्राई हाइब्रिड करते 1 to 1 2 4 2 1 to 1 then आपरे को 2 4 2 4 8 4 2 4 2 1 to 1 2 4 2 1 to 1 so the magic number is 1 to 1 2 4 2 1 to 1 एटा मन रखता तापरे नेक्स्ट क्वेश्चन को जिबा नले हमें किछ कर
2 4 2 1 to 1 2 4 2 1
Right, 1 to 1, 2, 4 to 1 to 1. Right. Now, then, अच्छा मु करिके आनिछि 150 टा क्वेश्चन टाइम हे गला रे आर थर को we will cover it as far as well मु कंसाइज कर देबी तमे टिके पढी के आस्तिबो अच्छा एटा गला नेक्स्ट क्वेश्चन ए ड्वार्फ पी प्लांट एटा पढ दियो मते आंसर टा को
The answer is C. All tall. All tall.
all tall
No, sir. Answer is 3:1.
3:1 हाँ डिफरेंस आसी गला हाँ तापरे नेक्स्ट
3:1.
3:1 आ गोटे आंसर 3:1 अच्छा बेसी समय नष्ट करबानी या को एनालाइज करबा किए करेक्ट दुई जण कहले 3:1 ना जणे कहले
All tall.
ऑल टॉल अच्छा जिबरलिन रो काम कोन
Growth.
जल्दी को
Growth hormone.
ग्रोथ जे कोन
It causes the increase in the height of the plant.
हां लेंथ रा बढ़ाई दिए जिबरलिन
Yes,
Yes, sir.
अच्छा ड्वार्फ प्लांट प्री प्लांट को तारो हमें जिबरलिन देले सेरा लंबा ही गला
Yes, sir.
Yes,
तो
sir.
तार जीनोटाइप ड्वार्फ थिला ना टॉल ही गला
Dwarf.
Dwarf.
रहिला अच्छा ड्वार्फ रहिले हमें जीनोटाइप ड्वार्फ एंड जीनोटाइप टॉल प्योर
Tall. Yes.
जीनोटाइप
Yes, sir.
टॉल एंड प्योर जीनोटाइप ड्वार्फ
Yes, sir.
ब्रीड कले कौन बाहरियो को
We'll get all tall plants.
जो माने 3:1 कहतलो बुझी पारलो
Yes, sir.
हां
Yes, sir.
डाउट रहिला जो माने 3:1 कहतलो
Yes,
तमे
sir.
माने नेक्स्ट ये को पळेलो इट इज जस्ट हाइब्रिडाइज करा हला हाइब्रिडाइज करा हले गोटे प्योर टॉल प्लांट आउ गोटे टॉल प्लांट जोटा जिबरलिन द्वारा हेची किंतु टॉल प्लांट रो जीन होची कैपिटल टी ड्वार्फ प्लांट रो जीन होची स्मॉल टी ए दुईटा मिसैले कोन बाहरिबो तार ऑफस्प्रिंग
Capital T, small t.
कैपिटल टी स्मॉल टी
Yes, sir.
Heterozygous tall.
सब गुडा को टॉल होबो काहे कि ना यारो गेमिट होची कैपिटल यारो गेमिट होची स्मॉल टी कैपिटल टी स्मॉल टी टुगेदर you will get capital T small t so it is hybrid tall so all will be tall so tall is the right answer next पढ़िकरी को आइटा कोन पढ़िकरी मत आंसर टाको
Sir, next screen.
Questions are not shared on the screen.
कर दो मूव हां right I'm sorry
and dwarf around in the yellow and the green character traits both characters direction
These are the traits.
Answer C, sir. Both
No, sir. These are the traits.
एवढा कैरेक्टर ना ट्रेट ना कैरेक्टर और ट्रेट जोटा ले भी हबो ना कोन ना जीनोटाइप
These are traits, sir.
sir, both C. C, sir.
see both
Both.
characters and traits अच्छा कैरेक्टर और ट्रेट भितरे किछि डिफरेंस अछि कि कैरेक्टर और ट्रेट भितर डिफरेंस टा कोन अछि कि तो अलगा कथा डिफरेंस टा कोन कैरेक्टर और ट्रेट भितर
Trait is a state of a character.
ट्रेट इज ए
State
हाँ.
of a character, which is a distinct variation of character.
राइट वेरी गुड ट्रेट माने height of the plant is a character, tall is a trait, dwarf is a trait, color of the seed is a character, round is a trait, wrinkled is a trait, yellow and green are traits and they are the color is the character. Any doubt? Print it in your mind. Character is the height, the color, the shape, the position, these are all characters. And what are traits? And 2-3 people are answered it as trait. It is trait. Trait means tall, dwarf, these things are traits. They are not characters. Height is the character. Okay, then next.
Three. Answer is B. Option B. 3.
Answer is B. What cross it is? Who cross item? Capital A, small a bane co cross. Monohybrid na dihybrid? Monohybrid. Monohybrid. Mane capital T, small t bhaliya, capital A, small a.
Yes.
Tahale genotypes कौन कौन हबो?
Capital A, capital A, capital A, small a, small a, small a.
Small a, small a. What is the ratio?
1:2:1.
1:2:1. ताको गुण देले केते आसला? 1:2:1, 2:4:2. 1:2:1. एटा कहार? डायहाइब्रिड. ताको प्रति गोटा को पुणी 1:4 रे गुण देले ट्राइहाइब्रिड आसीगला हला.
Yes.
With next पढ़ो.
Unit character.
Law of unit character.
Law of unit character. Next. How के कौन को?
Law of independent assortment.
How के कौन को जो? अच्छा मो यार रीजनिंग टा कहूची इंडिपेंडेंट असॉर्टमेंट माने कौन? इंडिपेंडेंट असॉर्टमेंट पाई कैरेक्टर गुडा को अलगा क्रोमोसोम रे रहबा दरकार ना सेम क्रोमोसोम रे रहबा दरकार? होमोलोगस क्रोमोसोम रे रहबा दरकार ना गोटिए क्रोमोसोम रे दीटा जाक रहबो?
Homologous chromosomes रे.
हाँ ताले गोटे क्रोमोसोम रे दीटा रहइबो ना रहइबोनी इंडिपेंडेंट असॉर्टमेंट रे? राउंड एंड रिंकल जदि गोटे क्रोमोसोम रे थिला ना अलगा क्रोमोसोम रे थिला?
अलगा थिला.
हां
होमोलोगस क्रोमोसोम रे रहइबो सर.
हां होमोलोगस क्रोमोसोम रे रहइबो जे मन करो जदि आमे दीटा कैरेक्टर जदि नबा दीटा कैरेक्टर नहले तो इंडिपेंडेंट असॉर्टमेंट हेइबोनी. राउंड रिंकल्ड येलो ग्रीन ए दीटा कैरेक्टर गोटिए क्रोमोसोम रे थिले ना दीटा क्रोमोसोम अलगा क्रोमोसोम रे थिले?
data chromosome.
एनीवन? हां दीटा अलगा क्रोमोसोम ना सेम क्रोमोसोम? होमोलोगस क्रोमोसोम जे
Different chromosome.
different chromosome. Kaiki na jodi same chromosome re rohile, it will become linkage. If they are on the same chromosome, it causes linkage. So linkage is against independent assortment. Independent assortment is possible only when the genes are present on separate chromosomes. But when there is linkage, they are present on the same chromosome. And if they are present on the same chromosome, then linkage is possible. It question kon kahila humko? Which law of Mendel would not have proposed if the phenomenon of linkage was known to him? Linkage jodi se jani thante, tahale se independent assortment kotha koi pari na thante. Se bhabi thante ita link ki kono. Acha, next karo.
Seven number of varieties, sir. Contrasting characteristics.
7. How can you answer?
Following number of varieties.
7, 14, 28 अनेक. 7. अच्छा. ताले दी तीन जनो कहले 7. Okay. Let us now come to the, you have given your assertion. Now we will come to the reasoning. अच्छा. Tall and dwarf. ये गुड़ा को trait ना character.
Traits.
Traits. Height of the stem is trait na character. Character. अच्छा. Mendel कितना character नहीं थे?
7 character.
7 character.
7 contrasting characteristics.
7 contrasting. ताहले तंकरो variety traits कितना हला?
14.
14, sir.
14. ताहले गोटे गोटे variety हबो ना नहीं?
Yes, sir.
Yes,
Yes
sir.
or no? हाँ तो वाला
Yes.
answer टा कौन हो?
14.
Answer 14. It is not 7. सेटिफाई मुझे क्वेश्चन टा बोलने बनेली. अच्छा, okay. Next.
Darwin, sir. Darwin.
डार्विन को पठाई नथिले नहीं? अच्छा, ओ. अच्छा, डार्विन आउ किए कौन कहूचो कि गेस मार के को? वाइल्ड गेस का मन को आछि को. ए नागुडा सुनी छ ना नहीं?
Sir, MJ Skeleton cell theory proposed.
Skeleton
cell theory proposed.
हाँ, cell
Darwin
theory
evolution रो.
Darwin evolution. नागेली? नागेली?
No, sir, no idea.
No idea. अच्छा. को हेले तमे कहूचो डार्विन किंबा जणा नहीं ना कोन ना 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 दियबो
Darwin.
डार्विन ओके एनीवन एल्स गेस मारो गाली करियर इति किछ नहीं एटा जान नथियो मु जानिछि
जाने न से
जानि न ताले एटा आंसर करिया पे तमे नेक्स्ट क्वेश्चन को आगे जा एटा भी जान नथियो
Sir. No.
जानिछो
No, sir.
नो यार आंसर हैला 40 40 मेंडल तकर ओरिजिनल पेपर रो 40 टा रिप्रिंट करथिले आउ ताको पठैथिले विभिन्न लोकों को
ना वो अबे जेबेन ट्रेनिंग कर साड़ी पिंदिबा
40
मोर पसंद साड़ी
हां कोनला
जाई कथा याद हां ना
आउ का
ना
सतो
उसरा जो वेट बसे मो गोटे मोबाइल सेट रखी छी तांके तो ला गोटे मोबाइल करिवे
प्लीज
ताको मोबाइल रे रखी छी ट्रेनिंग पे नेट करो तो
Please mute yourself.
Madam, please mute and discuss.
सर एक्सप्लेन करंतु टिके
हाँ 40 टा पेपर से करथिले से गुड़ा को रॉयल सोसाइटी को विभिन्न सोसाइटी को पठैथिले आ ता सहित बंद भी करूना
हेलो नमिता मैडम नमिता मैडम आपण म्यूट करंतु आपण हां सर कोंतु
हाँ at least I am happy that few are there who are very much interested but majority are busy in their own work अच्छा कौन हला हाँ से 40 टा पेपर रु डार्विन स्लेडन नागेली ए तीन जन को मध्य से पठाई थिले हला नागेली होचंती सेतबळर सबुठु नाम ज्यादा बॉटनिस्ट नागेली डार्विन तो फेमस नागेली भी वेरी फेमस डार्विन को भी से पठाई थिले हला नागेली स्लेडन डार्विन रॉयल बोटानिकल सोसाइटी सब आड़ को पठाई थिले केही तांकर पेपर को एक्सेप्ट करि पारले नाही 40 टा जगह को से पठाई थिले हला नेक्स्ट दिस इज फॉर योर नॉलेज नेक्स्ट अच्छा मुता आउट आंसर ता कहि दोछि केते जन को पठाई थिले से 40 हला ता भितरु डार्विन स्मार्ट नागेली समस्त को पठाई थिले अच्छा मते को तमे आंसर ता जानि छ क्वेश्चन भी आसीबो हु आर द पीपल नेक्स्ट क्वेश्चन ता को मेंडल्स कंट्रीब्यूशन के बाहर करथिले
ए करेन हुगो
डिवेरिस
एंड स्कावन
शेमार्क डिवराइस एंड कॉरेंस हला ए तीन जण अच्छा को वर्ष
1902
मैडम
1902
नो मेंडल वर्क एटा गोटे बेसिक मिस्टेक करछो बहुत बड़ा बोल मेंडल्स वर्क केबे रिडिस्कवर हेतिला
1856 to 1863
रीडिस्कवर केबे हेतिला एटा बेसिक क्वेश्चन एंट्रेंस एग्जामिनेशन मेंडल करो
In 1900.
1902 कहतिलो इट इज रॉन्ग इट इज 1900 अच्छा तीन जण करथिले डिवराइस चेमार्क एंड कॉरेंस हला तुमको केतरा इंटरेस्टिंग जिंस एटी कहूची नागेलिंग को कथा कोन कहिथिली मु प्रीवियस स्लाइड को गले बॉटनिस्ट सेतबळरो सबुटु बड़ बॉटनिस्ट नेक्स्ट जान अच्छा तांकर नागेलिंग स्टूडेंट होचंती कॉरेंस हला
यस सर
हां now Correns was the student of whom Kahar student?
Nageli.
Nageli student. Now, Chemark was the Chemark's father taught Mendel botany. Chemark's father was the teacher of Mendel. So out of these four, which two were indirectly or directly connected to Mendel? Correns. Because Correns was the student of Nageli, whom Mendel had sent his papers. Chemark was what? Was the son of a person who taught teacher. Now, say a question tapado. Which of the following was hesitant to recognize Mendel's contribution? से जानिले मेंडल आगु करचंती किंतु ताकू रिकॉग्नाइज करलेनी
Spillman.
was earlier done by Mendel, but Correns, who was a student of Nageli, he had seen Mendel's paper in Nageli's laboratory, and he recognized that this work was done by someone known as Mendel, and he warned De Vries. Se De Vries ku satarko kale je tome bhul korcho eta aguru heichi. Then De Vries recognized Mendel's work. And the fourth name that I have given there, please remember, he also rediscovered Mendel's work, Spillman. This is just for your remembrance. Spillman is an American who rediscovered Mendel's work in America. Others belong to Europe. You know that De Vries belongs to Netherlands or Holland. Chemark belongs to Austria, and Correns belongs to Germany. Now, besides these three, Spillman, who is an American, also recognized Mendel's work in 1901, 1 year later. Now, next question.
The answer is B, Darwin.
Darwin. This is correct. This is not a question where there is assertion and reasoning. This is a straightforward question, a straightforward answer. Whether you know it or don't know, that is a different thing. It depends on that. Next.
The allele gene are located on two homologous chromosomes.
They are located on homologous chromosomes. Right.
Two homologous chromosomes.
Homologous chromosomes. Right. Very good. Next.
Next please, sir.
Question action, sir.
जाओ नहीं, move करो. नहीं, आगू पड़ेगा. कोटा करेक्ट आंसर सब जगह मु पढ़ी परिबिनी तमे पढ़ी करी मते वो कोटा करेक्ट आंसर
Same height, flower color, flower position, pod shape, pod color, seed shape, seed color. Sir, A.
A, sir.
right
sir.
the answer is A. But never make a mistake to so hurriedly give the answer, because there may be a inherent misleading thing. What you said is correct.
Plant height, flower position.
अन्य गुड़ा टिके देखि दो just move your eye. Mane stem height, flower position, pod length. Eta nahi, so rejected. Then next are seed size nahi, so rejected. Then pod length outhare asti, rejected. So first is the right answer. Next.
नहीं है जो पुओ मानकर भी अच्छी देते वो तलटा पुओ मानकर
Due to incomplete or dominance.
Incomplete dominance.
Incomplete dominance.
Incomplete dominance. Anyone else? ଆଉ କିଏ କଣ କହିବାକୁ ଚାହୁଁଛ କି? ନା ଏଇଟା ମନକୁ ଆସୁ
ना
କୁହ।
कोई दियंतु सर
ମହରମ ନା କଣ ଥିଲା ବୋଧେ? ମହରମ ନା କଣ ଅଛି? ଆଜି ନା 23 କାଲି କଣ ହେଇପାରୁନି? ଏଟା ମୋ ମତରେ ସବୁଠୁ ସହଜ ଆନ୍ସର।
Linkage, sir.
Anyone else? प्रथमे
Complete dominance.
Complete dominance.
एटा बसे दियंतु
Incomplete dominance. Complete dominance. Linkage. कहि दोछि मु आंसर.
complete dominance हबनी
Complete dominance हबो. अच्छा.
हबनी
सुनो प्रथमे जने कहले जे it is incomplete dominance. अच्छा, incomplete dominance रे red and white crossed together gave us pink. Question रे कोन लिखा है? Parental types are greater than recombinants.
Yes.
When the F2 generation, hella F2 generation ratio kono incomplete dominance ro incomplete dominance ro ratio kono
1:2:1.
1:2:1.
Yes, sir.
ताले ये 1:2:1 रो parental combination कोटा non parental कोटा
Parental combination sir 2 hobo 1 purely tall, 1 purely
parental combination
and
2 hobo parental combination 2 hobo
1 sir.
1 parental combination जति 1 now 1 हैला 2 टा कोन non parental ना नहीं
Sir
2 टा होची non parental pink 2 टा होची pink 1 is red 1 is white. So which is greater, parental or non parental? Non parental is greater.
non parental
So incomplete dominance is wrong. Now
Yes.
complete dominance जति हल्ला जे complete dominance answer कहला complete dominance हले सब गुड़ा का गोटे प्रकार बाहरियो ना नहीं tall and dwarf cross together only tall किछिटा जदि recombination माने नुआ बाहरुची आउ बहुत गुड़ाए ओरिजिनल रोची ताले का द्वारा linkage hale they are not on they are not on separate chromosomes. The two characters are not on separate chromosomes, they are on the same chromosome. So there is linkage. Because linkage is there, they tend to move together. ସେମାନେ ଏକାଠି ଯିବାକୁ ଚେଷ୍ଟା କରିବେ ଏକାଠି ଗଲେ କଣ ହବ ତାଙ୍କର ନମ୍ବରଟା ବଢିବ ସେମାନେ କେତେବେଳେ ଅଲଗା ହେବେ ଲିଙ୍କେଜ ରେ କେବେ ଅଲଗା ହୁଅନ୍ତି ମତେ କହିଲ लिंकेज केतेବେଳେ अलगा हुए
during distance in between two present on a chromosome.
it is by crossing over linked genes are separated due to crossing over crossing over जेतिकी हबो सेतिकी नुवा combinations बाहरिबे माने recombination बाहरिबे सेति पई because of linkage the recombinants are very less. Now it is almost time. It is almost time. So next time my sincere request is
Come prepared.
Come prepared. Now I tell you, you all run after modern biotechnology, molecular biology, this thing, that thing, but basic things are these. Now if these you do not know, then there is no use. So these things also you must know. You give importance to that, but you must learn these things also. Now, next class, we will continue with this, and I will prepare some more questions, and we will discuss in the next class when we meet. Thank you very much.
Thank you. Thank you. We now come to the end of this today's program. So our next class, sometime after 7 or 8 days, I've given you the timetable. We'll join on that particular day. But while attending the classes, be sincere and try to interact with the research persons. Because this is a new pattern and you have to hand it over to the students. So come prepared for the next class and the topic. Okay, all the best. Best of luck. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Yes, sir.